Welcome to the deep dive. Today, we're embarking on really an extraordinary journey deep into one of the most evolutionarily conserved and, well, utterly essential regions of your brain, the hypothalamus. Imagine it as your body's ultimate control center, kind of orchestrating everything from your hormones and your sleep to your appetite, and even those fundamental emotional responses. To truly grasp what makes us tick, this is really where we begin.
Right. And what's truly fascinating about the hypothalamus is how this, really small collection of neurons, often called the brain of the brain, acts as the critical bridge between your nervous system and your endocrine system. It's constantly working behind the scenes to maintain your body's delicate balance, a really complex symphony of feedback loops. Understanding it is, I think, truly a shortcut to being deeply informed about the very core of your physiological well-being. It lets you master this intricate information, like the elite professionals in the field aim to do.
We've certainly got a dense stack of sources
Yeah.
for this deep dive. Yeah, from foundational textbooks to really cutting edge research. And our mission today is to unpack every important nugget of knowledge and insight contained within them. We're going to explore every topic, every concept, so you can really get a handle on this information, truly master it. So let's begin our deep dive into the intricate world of neuroendocrinology. Okay, let's actually start with something maybe a little surprising, a historical fact. The connection between the hypothalamus and the pituitary gland was recognized astonishingly early. We're talking all the way back in the 2nd century AD by Galen of Pergamon.
That's right, Galen. In his treatises, the Usu Partium and Anatomicae Administrationes, he described a physical link. He talked about the infundibulum, that's the stalk connecting the hypothalamus to the pituitary, and a surrounding vascular network, which he called the tet mirabilis, or the wonderful net. But his understanding was, well, quite different from ours today. He viewed the pituitary, which, literally means phlegm in Latin, pituita. He thought it was a filter for brain waste. That's where the gland actually got its name.
Huh, brain waste. Okay. And obviously, as we fast forward to modern times, the field of neuroendocrinology has clearly expanded dramatically, far beyond just controlling the pituitary.
Oh, absolutely. If you connect this to the bigger picture, neuroendocrinology now really encompasses the complex reciprocal interactions between the central nervous system, the CNS, and your endocrine systems. These interactions are crucial not just for maintaining your body's internal stability, homeostasis, but also for responding effectively to every environmental stimulus you encounter. Think about the discovery of hormones like leptin back in 1994. That profoundly affects multiple neuroendocrine circuits, like, for instance, suppressing thyroid and reproductive axes during starvation, or ghrelin, that stomach peptide regulating appetite. Discoveries like these truly showcase just how much more there was, and frankly still is, to learn.
Yeah, and I imagine studying these tiny diffuse neurons must have been historically a huge challenge for researchers, just physically finding and analyzing them.
It absolutely was.
Yeah.
Which, naturally leads us to wonder, how do we study such small quantities of neurons and their complex interactions today? Transformative technologies have really revolutionized our capabilities. For example, researchers now use transgenic mice. These are mice engineered to express fluorescent marker proteins in specific hormone-producing neurons, like those for GnRH or POMC. We can also use viral vectors to precisely modulate or change the activity of specific cells. These tools let us study these hypothalamic neurons both in vitro, in slice preparations, in a controlled lab environment, and also in vivo, within living organisms.
So ultimately, in neuroendocrinology, it really boils down to that fundamental interaction between the nervous system and the endocrine system.
Precisely. It's the deep study of how these two powerful systems work together. Not just in those classic, well-known axes like reproduction or growth, but also in broader homeostatic systems like energy balance and immune function. In these broader systems, those classic axes are important, sure, but they're definitely not autonomous, not independent pathways. It's all interconnected.
And at its very core, neuroendocrinology involves these specialized neurons secreting signaling molecules, hormones, directly into circulation.
Yeah, a fundamental principle guiding this whole field is the highly regulated secretion of hormones, neurotransmitters, or neuromodulators. Both endocrine cells, those that release hormones into the blood, and neurons are, well, prototypical secretory cells. What this means is they both have electrically excitable plasma membranes and specific ion conductances that regulate the exocytosis. That's the outward release of their signaling molecules from storage vesicles.
Okay, so how do these different cells actually release their contents into the body? How do they, differ in their approach?
Right, we classify them based on their topographic mechanisms, essentially where they secrete their contents. So you have endocrine cells, They secrete directly into the bloodstream, acting globally as hormones, influencing distant parts of the body. Then there are paracrine cells. They release their contents into the extracellular space, affecting only closely neighboring cells. It's more of a local communication. You also have autocrine cells, which affect their own function through the local actions of their own secretions. They're kind of talking to themselves, in a way. And then in contrast, you have exocrine glands. These secrete proteinaceous substances like enzymes and lipids into the lumen of the ductal systems. So they release their products into ducts or tubes, not directly into the bloodstream.
That's really fascinating. And it's not just the brain directly controlling the endocrine system. The autonomic nervous system actually plays this huge, often kind of hidden role, too.
That's a major precept, yeah. Key idea in neuroendocrinology. The nervous system, particularly the autonomic nervous system, which controls all your involuntary bodily functions, it controls or modifies the function of both endocrine and exocrine glands. While the anterior pituitary is exquisitely controlled by these releasing factor hormones from the hypothalamus, other glands like the pancreas, the adrenal glands, the pineal gland, even salivary glands, are directly regulated by cholinergic and noradrenergic inputs from the autonomic nervous system.
And this interconnectedness clearly extends to really critical metabolic processes within the body, like managing glucose.
What's particularly fascinating here is how the autonomic regulation of glucose homeostasis is modulated by these interactions between your gut and your hypothalamus, and even by substances derived from adipose tissue, fat like leptin. For instance, the hypothalamus can directly sense bile acids. These acids, after they're released into the small intestine, stimulate the secretion of something called fibroblast growth factor 19, or FGF19. This FGF19 then acts on specific neurons in the mediobasal hypothalamus to induce autonomic responses that ultimately lower blood glucose via a complex multisynaptic pathway. Similarly, changes in gut osmolality, that's the concentration of solutes in your gut fluids, transmit a gut-to-brain signal through the vagus nerve, contributing to that feeling of thirst satiation, when you feel like you've had enough to drink. Understanding this complex interplay is absolutely essential for diagnosing and effectively managing endocrine disorders like diabetes mellitus.
Okay, let's really zoom in now on the hypothalamus itself. It's often called the ultimate brain structure for maintaining homeostasis. And its destruction, well, tragically, it's just not compatible with life. It's that vital.
That's profoundly true. Its comprehensive control stems from its ability to orchestrate these coordinated endocrine, autonomic, and behavioral responses. Think of it like a central hub, constantly receiving sensory inputs from the external environment, light, pain, temperature, smells, and crucial vital information about your internal environment, like blood pressure, osmolality, glucose levels. And what's more, hormones like glucocorticoids, gonadal steroids, thyroid hormone, and leptin directly exert both negative and positive feedback on the hypothalamus, constantly updating its internal state, telling it what's going on.
So it really takes all this diverse information in, processes it, and then sends out this whole symphony of finely tuned responses.
Exactly. The hypothalamus integrates these diverse inputs and then directs coordinated motor outputs to key regulatory sites throughout your body. These include the anterior and posterior pituitary glands, of course, but also the cerebral cortex, brainstem, and spinal cord motor neurons, limbic system structures like the amygdala, septum, hippocampus, thalamic nuclei, all crucial for emotion and memory, and also to parasympathetic and sympathetic preganglionic neurons that control your automatic bodily functions. This intricate network ultimately results in the coordinated endocrine, behavioral, and autonomic responses essential for maintaining your homeostasis day in, day out.
Right. And a crucial part of this entire unit is the posterior pituitary, which is also known as the neural lobe.
Indeed. The major component of the neural lobe consists of axon terminals, the ends of nerve fibers, originating from what we call magnocellular secretory neurons. These are large neurons located in two specific nuclei of the hypothalamus, the paraventricular nucleus, PVH, and the supraoptic nucleus, SON. These terminals are closely associated with a capillary plexus, a dense network of tiny blood vessels, and they directly secrete substances like arginine, vasopressin, AVP, also known as ADH, and oxytocin into the hypophyseal veins and then into the general circulation. The blood supply to this area comes from the inferior hypophyseal artery, which is a branch of the internal carotid artery.
And it sounds like there are special cells in there, too, that play a unique role in helping regulate this whole process.
There certainly are. Glial-like cells, which are supportive cells in the nervous system, called pituocytes, they make up about 25% of the posterior pituitary, and they play an important role in regulating vasopressin and oxytocin secretion through at least two mechanisms. First, through structural modifications. They can actually engulf or retract from the neurosecretory axon endings, physically changing how much hormone can get out. Second, they release hormone modulators, for example, galanin-like peptide, GALP, which stimulates release, or taurine, which inhibits the release of vasopressin. So ultimately, the PVH, the S on N, and their axon terminals down in the neural lobe are the main effector arms for centrally regulating your blood osmolality, your fluid balance, and your blood pressure.
Okay, so oxytocin. We often hear about it in the context of childbirth and milk letdown, but you mentioned it does much more beyond those familiar rules.
It absolutely does. Its secretion by those magnocellular neurons is undeniably critical at parturition, childbirth, for uterine myometrial contraction. And it's famously regulated by the classic milk letdown reflex. Mechanosensory information from the nipple reaches these neurons, causing a synchronized burst of action potentials and oxytocin release, which then leads to milk release. However, oxytocin also acts on many other brain regions, including the medial preoptic area, the ventral tegmental area, the nucleus accumbens, and the amygdala. These actions influence synaptic plasticity, how our brain adapts and learns, and profoundly affect social behavior, particularly parental behavior, by reducing stress and increasing empathy. Interestingly, while vasopressin might mediate various forms of aggression, maternal defensive aggression toward intruders also involves oxytocin. And it's worth noting that vasopressin also synergizes or works cooperatively with CRH, the stress hormone.
Now, let's not overlook the median eminence. It's a tiny area, but you said it's incredibly vital to this system.
The median eminence holds an essential role in regulating the pars distalis, which is the anterior pituitary. Its unique anatomical location positions it as both an afferent sensory organ, meaning it receives sensory information, and a crucial functional link between the hypothalamus and the pituitary. It's directly adjacent to several important neuroendocrine and autonomic regulatory nuclei within the hypothalamus, including the infundibular, also called arcuate, ventromedial, dorsomedial, and paraventricular nuclei. It's right there in the middle of the action.
And within the median eminence itself, there are these really intriguing glial-like cells, the tanycytes, with specialized functions.
Absolutely. The external zone of the median eminence contains these specialized tanycytes. There are glial-like cells involved in nutrient sensing, energy homeostasis, and surprisingly, they even function as stem or progenitor cells for neurogenesis, the creation of new neurons in gliogenesis, the creation of new glial cells, even in adult animals. The internal zone, on the other hand, is primarily composed of axons from those magnocellular neurons of the axon and PVH. These axons contain characteristic large neurosecretory granules, often called herring bodies, which are basically stored hormone packages on their way down to the posterior pituitary. It also includes axons of hypophysiotropic neurons and candy-site processes destined for the external layer, forming this really complex interface.
Okay, then there's the pars tuberalis, which just sounds a bit mysterious based on its name.
It is a bit mysterious. The pars tuberalis is a subdivision of the adenohypophysis, or anterior pituitary. It's basically a thin sheet of glandular tissue that surrounds the infundibulum and the pituitary stalk. While its precise physiological role in humans remains somewhat enigmatic, it clearly participates in the CNS's photosensing pathway. It expresses high concentrations of the melanocortin-1 receptor. In some mammals and birds, it's definitively established to regulate seasonal reproduction, energy balance, and body weight, primarily through melatonin release. It also secretes other substances, including neurokinin A, substance P, and endocannabinoids, which influence seasonal prolactin release in some species through direct effects on the pars distalis. So it's doing something important even if we don't fully grasp its human role yet.
Right. Now, our brains are so incredibly protected by the blood-brain barrier. So how then do they manage to sense things like hormone levels or toxins that might be circulating in the bloodstream? It seems like a bit of a contradiction.
That's a crucial point, and a guiding principle of neurophysiology. The brain truly resides in an environment protected by this blood-brain barrier. This barrier, formed by tight junctions between brain vascular endothelial cells, astrocyte foot processes, and perivascular microglia, prevents the free passage of large molecules. However, to exert its vital homeostatic control, the brain absolutely needs to assess key sensory information from the bloodstream hormone levels, metabolites, potential toxins. This is precisely where what we call windows on the circulation come in. These are the circumventricular organs, or CVOs.
So what specifically makes these CVOs different? How do they actually bypass that protective barrier?
What makes them unique is that they lack a true blood-brain barrier. Instead, they possess fenestrated capillaries. That means capillaries with small pores or windows in their walls that allow for the relatively free passage of substances directly from the blood into the surrounding brain tissue. CVOs like the median eminence, the organ vasculosum of the lamina terminalis, OVLT, and the subfornical organs serve as these critical links. They transmit peripheral metabolic cues, hormones, and even potential toxins directly to key neuronal cell groups in the brain. These neurons then regulate coordinated endocrine, autonomic, and behavioral responses. For example, the OVLT projects to many brain regions and is deeply involved in diverse processes, including, as we mentioned, thirst satiation. They're essential sensing points. Okay, moving on. Descartes famously called the pineal gland the seat of the soul. Well, that's certainly poetic. What's the modern scientific view of its role? Yeah, a more contemporary, though maybe less colorful, viewpoint is that the pineal gland integrates information encoded by light into coordinated secretions that ultimately underlie biological rhythmicity. It's quite unique because it functions as both an endocrine gland, releasing hormones, and a circumventricular organ, meaning it has that direct window to the blood we just talked about. It's derived from cells in the roof of the third ventricle of the brain.
And of course, it's primarily known for secreting the hormone melatonin.
Yes, the predominant hormone secreted by the pineal gland is indeed melatonin. However, it also contains other biogenic amines, various peptides, GABA, and even neurosteroids, like allopregnanolone. Allopregnanolone is important because it prevents neuronal apoptosis or programmed cell death and also influences locomotor activity. Melatonin itself is synthesized from the amino acid tryptophan through a series of enzymatic steps. A key enzyme, arylocalamine and acetyltransferase, or ANET, catalyzes the rate-limiting step, producing N-acetylserotonin. This is then followed by another enzyme, hydroxyindolemethyltransferase, HIOMT, which adds a methyl group to create melatonin.
So, given that enzymatic pathway, how exactly is melatonin synthesis so precisely regulated within the body? It seems very tied to light.
It is. A fundamental principle of circadian biology is that melatonin synthesis is exquisitely controlled and trained synchronized by the light-dark cycle, with darkness being the most important signal. ANET messenger RNA levels, ANET enzyme activity, and consequently melatonin synthesis and release are all highest during the dark period and decrease sharply with the onset of light. Melatonin isn't stored significantly. It's released into the blood or cerebrospinal fluid, CSF, pretty much directly after biosynthesis, in proportion to ANET activity. However, it has a very rapid half-life, just minutes due to rapid metabolism, mainly in the liver.
So the presence, or crucially the lack of light, is the key trigger for its release.
Precisely. A lack of light ultimately results in the release of norepinephrine from sympathetic nerve terminals. These act on beta-adrenergic receptors in pinealocytes, the cells of the pineal gland. This interaction increases adenylyl cyclase activity, leading to cyclic adenosine monophosphate, CMP synthesis. Increased CAMP then activates downstream signaling pathways, including protein kinase A and the phosphorylation of CAMP response element binding protein, or SERB. CREB acts directly on the NAT promoter, increasing its expression. So the light-dark cycle, specifically acting through the sympathetic nervous system, dramatically changes melatonin levels across the day. It's important to note, though, that human melatonin regulation differs a bit from rodents. We humans require significantly more light to suppress nocturnal melatonin, and NAT control is largely post-transcriptional in our species, meaning the regulation happens after the gene is transcribed into RNA.
Melatonin clearly helps us keep time, but what's the actual master clock in our brain that sets all these rhythms?
The best understood neural structures responsible for orchestrating our circadian rhythms, those roughly 24-hour cycles, are the suprachiasmatic nuclei, or SCNs. These are paired structures located in the anterior hypothalamus, directly above the optic chiasm where your optic nerves cross. These SCNs truly function as your central circadian clock, orchestrating multiple biological rhythms throughout your entire body. What's remarkable is that individual cells within the SCN, including both neurons and astrocytes, possess an intrinsic capacity to oscillate in a circadian pattern. This is due to a cell-autonomous transcription-translation feedback loop. It involves positive regulators like CLOCK and BML1, which interact with the promoters of the negative regulators, the period, PER, and cryptochrome, CRY genes, creating this elegant molecular pendulum that keeps time.
And what are the consequences if this finely tuned biological clock, the SCN, gets disrupted, say through injury or disease?
Well, if the SCN is lesion bilaterally damaged on both sides, it produces what's called free-running circadian rhythmicity. This is characterized by a severely disrupted sleep-wake cycle and a loss of predictable daily oscillations in things like feeding, drinking, melatonin secretion, and the secretion of some anterior pituitary hormones. But what's truly remarkable and really highlights its role as the master pacemaker is that normal rhythmicity can actually be restored if SCN tissue is transplanted back into the lesioned animals.
Wow. The SCN sounds incredibly complex given all the inputs it receives and the outputs it controls.
It is indeed. The SCN has two major subdivisions, a dorsal shell and a ventral core, and each receives different inputs, making its integration really sophisticated. The core receives direct light signals via the retinohypothalamic projection, carrying signaling molecules like pituitary adenyl cyclase activating peptide, PACAP, and nitric oxide. It also gets neuronal input from GABA-containing a neuropeptide Y, MPY, containing axonal projections from the intergeniculate leaflet of the thalamus, as well as serotonin neurons from the midbrain raphe. The shell, meanwhile, receives input from the limbic system structures like the hippocampus, the bed nucleus of the stria terminalis BNST, and the septum and the hypothalamus itself. These diverse inputs are crucial for modulating the endogenous rhythms of individual SCN pacemaker cells and contribute to phase-shifting effects on SCN activity. The SCN is also rich in reciprocal neuron interactions mediated by GABA and contains numerous neuropeptides like AVP, VIP, gastrin-releasing peptide, GRP, and calretinin, all vital for synchronizing its cellular rhythms. And of course, it directly responds to melatonin through its own melatonin receptors, completing a feedback loop.
Okay, beyond just sleep, it seems melatonin has some pretty surprising effects, especially as you mentioned on reproduction.
Yeah, what's fascinating here is that one of melatonin's best characterized roles is its regulation of the reproductive axis. This includes gonadotropin secretion and the precise timing of puberty, which is particularly prominent in seasonal breeders like rodents and sheep. Classic experiments have clearly shown that pineal gland removal in these animals leads to precocious or early puberty. Conversely, constant darkness or blindness results in testicular atrophy and decreased testosterone, a condition that could then be normalized by removing the pineal gland. This reproductive strategy likely evolved to synchronize gestation with favorable birth times, ensuring offspring are born when resources are most abundant.
So, how precisely does melatonin signal this photoperiodic information, the length of day, to the reproductive axis? What's the pathway?
It's an intricate signaling pathway. It involves changes in the nocturnal duration of melatonin, conveying photoperiodic information to cells in the pars tuberalis via the MT1 receptor. This then regulates clockwork proteins like Cry1, which are linked to TSH synthesis. TSH, acting as a paracrine signal acting locally from the pars tuberalis, binds to TSH receptors on tanycytes. Remember those specialized glial cells? Their end-feet processes reside immediately dorsal to the pars tuberalis, perfectly placed to receive this signal. These canycytes then regulate the expression of type 2, D2, and type 3, D3, iodothyronine deiodinase. These are enzymes that either activate or inactivate thyroid hormone, respectively. This mechanism governs thyroid hormone's bioavailability right there in that crucial hypothalamic region. So under long day conditions, shorter melatonin duration D2 is upregulated, increasing active T3 and activating reproduction. Conversely, under short day conditions, longer melatonin duration D3 is
Wow, so what you're saying is that local thyroid hormone levels controlled by this pathway are absolutely crucial for precise reproductive timing in these animals.
absolutely. An increase in T3 bioavailability results directly in reproductive activation. And this effect can actually be replicated by implanting a T3 pellet directly into the hypothalamus of seasonal breeders kept in short days, which would normally shut down reproduction. Thyroid hormone also affects GnRH secretion by regulating the physical encasement of GnRH axon terminals in the external zone of the median eminence by those tanycyte end-foot processes. When thyroid hormone is administered into the hypothalamus, it promotes the retraction of these processes, allowing direct access of secreted GnRH to the fenestrated portal vessels for transport to the pituitary. Thyroid hormone receptors are also plentiful throughout the hypothalamus and may specifically regulate kisspeptin, that hormone critical for pulsatile GnRH release we'll talk more about.
Okay, let's quickly touch on melatonin receptors again and its various therapeutic uses in humans.
Right. Melatonin mediates its effects through a family of G-protein-coupled receptors, GPCRs, specifically MT1 and MT2. There's a third receptor in mammals, MT3, but it's not a GPCR. It's actually a high-affinity binding site on a cytosolic enzyme called quinone reductase 2, which is involved in cellular detoxification. This might help explain some of melatonin's well-known antioxidant effects, as melatonin also directly acts as a free radical scavenger, mopping up reactive oxygen and nitrogen species.
And for human therapy, how is melatonin primarily used and what are its most established benefits?
Well, while melatonin is often purported to have many beneficial functions, things like anti-aging effects, blood pressure regulation, neuroprotection in diseases like Alzheimer's and Parkinson's, even immune enhancement, its most extensively studied and truly established role in humans is phase shifting and resetting circadian rhythms. It's widely used to treat jet lag and may be effective for various circadian-based sleep disorders. Now, while studies do show it can regulate sleep, meta-analyses suggest the evidence for significant clinical efficacy for primary or secondary sleep disorders is somewhat limited, although it is widely considered safe for short-term use, typically up to about 3 months. It has also been suggested to have oncostatic effects on some tumors, reduce blood pressure and hypertension, improve symptoms of major depressive disorder and anxiety, and exert anti-inflammatory actions. So its clinical potential is quite broad, even if some areas definitely require more definitive research to confirm the benefits.
Okay, now moving to the real core of this system. The hypothalamus controls the pituitary by releasing these specific hormones directly into the portal circulation, acting like a master conductor for the endocrine orchestra.
That's right. And the search for these releasing factors, as they were initially called, was a truly Herculean effort back in the day. I mean, consider this. Approximately 250,000 hypothalamic fragments were required just to purify and characterize the very first one, thyrotropin-releasing hormone, PRH. These substances are now more commonly known as releasing hormones, reflecting their critical endocrine function.
Are all of these releasing hormones peptides then? Or are there exceptions?
Most are peptides, which are short chains of amino acids. But there's one notable exception, dopamine. Dopamine is a biogenic eminy, and it serves as the major prolactin inhibiting factor, or PIF. Many of these hypothalamic hormones, along with their therapeutic analogs, synthetic versions, are widely available now for clinical investigations or diagnostic tests, like those for dopamine, GnRH, and somatostatin.
And it's clear they have incredibly complex interactions, right? It's
Yeah.
not just simple one-to-one effects on the pituitary.
It's much more nuanced. Beyond simply regulating hormone release, some hypophysiotropic factors also control the differentiation of pituitary cells into specific types, their proliferation, their growth, and even their ability to synthesize hormones. And many of these factors actually act on more than one pituitary hormone. For instance, TRH is a potent releaser of both prolactin, PRL, and thyroid-stimulating hormone, TSH, and sometimes even corticotropin, ACTH, and growth hormone, GH. GnRH releases both luteinizing hormone, LH, and follicle-stimulating hormone, FSH. Somatostatin, the master inhibitor, blocks GH, TSH, and a wide variety of non-pituitary hormones. And dopamine, the principal inhibitor of PRL secretion, also inhibits TSH and gonadotropin, and under certain conditions, GH. This truly demonstrates a sophisticated dual control system involving both inhibitory and stimulatory hypothalamic hormones. For example, somatostatin interacts with GHRH and TRH to precisely control GH and TSH secretion, respectively. Dopamine interacts with prolactin-releasing factors, PRS, to regulate PRL secretion. Some hormones even act synergistically, like CRH and AVP working together to regulate pituitary ACTH release. It's layers upon layers of control.
This sounds less like simple biology and much more like a highly sophisticated engineering control system.
It absolutely resembles complex engineering systems. You can think of it having a sensing element that detects the hormone concentration, a reference input that defines the desired control levels, and an error signal that determines the system's output to adjust for any deviation. The precise set point of the controlled variable, like your thyroid hormone level, is determined by a complex cascade involving receptor-binding kinetics and successive intermediate messengers within the cells. Sophisticated models incorporating these control elements, compartmental analysis, hormone production clearance rates, often referred to as systems biology, exist for many of these systems, further emphasizing their intricate design.
Our bodies certainly seem to run on a very precise internal clock, wouldn't you say? These hormonal rhythms.
They absolutely do. Virtually all functions of living animals are subject to periodic or cyclic changes, and many of these are primarily influenced by the nervous system. Most of these periodic changes are free-running, meaning they're intrinsic to the organism, driven by an internal biological clock independent of the immediate environment. However, these free-running rhythms are then coordinated or entrained by external signals. We call these zeitgivers or time givers, things like light-dark changes, meal patterns, even lunar cycles in some species. Many endogenous rhythms have a period of approximately 24 hours, and these are of course known as circadian rhythms.
Why does this precise timing and rhythmicity matter so much clinically, in terms of diagnosing and managing health conditions?
Well, the assessment of endocrine function absolutely must account for the inherent variability of hormone levels throughout the day or night. Appropriately obtained samples at different times can provide incredibly useful dynamic indicators of hypothalamic pituitary function. For example, the loss of the normal diurnal rhythm, that daily fluctuation of GH and ACTH secretion, can be an early and sensitive sign of hypothalamic dysfunction. Furthermore, the optimal timing for administering medications like glucocorticoids to suppress ACTH secretion in therapies for congenital adrenal hyperplasia has to consider the axis's varying suppressibility at different times of day. You can't just give it any time. It's all about hitting the right window.
Okay, let's talk about thyrotropin-releasing hormone, TRH. Obviously vital for thyroid function, but it appears to have a much broader evolutionary and functional story than just that single role.
It's true. TRH is phylogenetically ancient, meaning it's found even in primitive vertebrates and invertebrates. Now, while it doesn't stimulate TSH release in all those species, its wide expression suggests it was co-opted or adapted as a hypophysiotropic factor midway through vertebrate evolution, perhaps specifically for coordinated temperature homeostasis. Although the TRH tripeptide, that small molecule of 3 amino acids, is the only established hormone derived from its prohormone, the rat pro-TRH actually yields 7 additional peptides with unique tissue distributions and biological activities, suggesting a much wider biological scope than just thyroid control.
So at the cellular level, how does TRH exert its diverse effects once it's released and binds to its target?
Right. Stimulatory effects begin with TRH binding to its G protein-coupled receptor, its GPCR, on the thyrotroph plasma membrane. Those are the pituitary cells that produce TSH. Interestingly, thyroid hormone and somatostatin antagonize or counteract these effects without actually interfering with the TRH binding itself. They work downstream. TRH action is mainly mediated through the GQ11 protein coupled to the type 1 TRH receptor. This leads to phosphatidylysyl hydrolysis, the phosphorylation of key protein kinases, and a crucial increase in intracellular free calcium. This calcium increase is fundamental for post-receptor activation. TRH not only stimulates the formation of messenger RNAs coding for TSH and prolactin, PRL, but it also regulates TSH glycosylation, the addition of sugar molecules, and secretion, and even stimulates thyrotroph mitogenesis, which is the proliferation or growth of these TSH-producing cells.
And it's really fascinating that TRH isn't just found and active in the hypothalamus controlling the pituitary. Its reach is far wider.
Absolutely. Beyond those specific hypophysiotropic TRH neurons in the hypothalamus, TRH is also synthesized by non-hypophysiotropic neurons throughout the brain. This includes many other neuronal groups within the hypothalamus itself, as well as the amygdala, hippocampus, thalamus, brainstem, and spinal cord. Its extensive distribution and the widespread presence of TRH receptors suggest it functions broadly as a neurotransmitter or neuromodulator across the central nervous system. Centrally administered TRH has diverse effects. It reduces appetite, increases locomotor activity, reduces anxiety, has anti-epileptic effects, and can induce hyperthermia, indicating a vital role in central thermoregulation. It also broadly affects autonomic regulation, increasing blood pressure, heart rate, gastrointestinal motility, gastric acid secretion, and even insulin secretion. Additionally, it acts as an excitatory modulator of lower motor neuron function. You can even find TRH outside the brain in places like anterior pituitary somatotrophs, the retina, thyroid parafollicular cells, adrenal medulla, and various reproductive and digestive organs. It's truly a versatile molecule.
Given that incredibly broad range of effects, TRH sounds like it could have immense therapeutic potential for a lot of different conditions.
It absolutely has. TRH has been extensively evaluated for diverse neurobiologic disorders, including spinal muscle atrophy and amyotrophic lateral sclerosis, ALS. Now, while transient improvements in strength have been reported, long-term efficacy hasn't yet been definitively confirmed. TRH administration also significantly reduces the severity of experimentally induced spinal and ischemic shock and shows promise in improving recovery after spinal cord injury and head trauma. It has also been used to treat children with complex neurologic disorders like West syndrome and intractable epilepsy. So its potential in these areas remains a very active field of research.
Let's clarify its crucial feedback role with thyroid hormones. How do they interact so precisely to maintain balance?
Right, the feedback is key. Thyroid hormones, specifically T3 and T4, act on both the pituitary and the hypothalamus, exhibiting remarkably precise feedback control. Even small doses of T3 and T4 inhibit the TSH response to TRH, and barely detectable decreases in plasma thyroid hormone levels actually sensitize the pituitary to TRH, making it more responsive. Now, while TRH acts quickly via its GPCR on the cell surface, thyroid hormone actions, which are mediated by intranuclear receptors inside the cell nucleus, require several hours to manifest their effects. This negative feedback of thyroid hormone on hypophysiotropic TRH neurons is mediated by a specific thyroid hormone receptor, TR2, which is expressed in these neurons within the paraventricular nucleus, or PVH. Interestingly, T3 alone isn't sufficient for this feedback. T4 is needed. It's likely converted to the more active T3 by the D2 enzyme within tanycytes, which then transports T3 to TRH neurons, inhibiting TRH via retrograde axonal transport, essentially signaling back up the axon.
What about the impact of external factors like stress and nutrition on TRH regulation? How do they fit in?
Catecholamines, released from brainstem neurons, can rapidly increase TRH release, affecting both TRH cell bodies in the PVH and their axon terminals down in the median eminence. Stress is another significant determinant of TSH secretion. In humans, physical stress like intense illness or trauma typically inhibits TSH release. This is why low T3 and T4 levels observed in non-thyroidal illness syndrome don't cause the compensatory TSH increases you'd normally expect. Glucocorticoids, our stress hormones, generally blunt the thyroid axis through actions within the CNS, and the TRH gene itself even contains a glucocorticoid response element, suggesting direct influence. Now, while glucocorticoids can stimulate TRH production in isolated experimental settings, their overall inhibitory effect on the thyroid axis usually results from indirect negative feedback on structures like the hippocampus. This disruption of hippocampal suppression of the HPA axis is, in fact, linked to hypercortisolemia, high cortisol levels, in affective illnesses like depression. What's particularly interesting, maybe counterintuitive, is how the body responds to dehydration-induced anorexia, or DIA, where food intake is markedly reduced. Despite this, TRH gene expression in the PVH actually increases rather than decreases. This mechanism might involve activating effects of CRH from lateral hypothalamic neurons, which project to hypophysiotropic TRH neurons and are known to reduce TRH release. It shows a very complex interplay depending on the situation.
Okay, let's shift to corticotropin-releasing hormone, or CRH. This is widely recognized as the central player in our body's stress response, the HPA axis.
The HPA axis is indeed the humoral or fluid-borne component of an integrated neural and endocrine system. It's vital for regulating energy homeostasis through its effects on hepatic glycogenolysis, the breakdown of liver glycogen, and lipolysis, the breakdown of fats. It truly is your body's primary response mechanism to both acute and chronic internal and external stressors. This system coordinates intimately with the autonomic nervous system to induce that well-known fight-or-flight response via catecholamines released from the adrenal medulla. Pituitary ACTH secretion is primarily stimulated by CRH, which originates from parvocellular neurons, small neurons in the PVH. To a lesser extent, it's also stimulated by AVP, which can actually be co-secreted from these same hypophysiotropic CRH neurons. And beyond its direct pituitary action, the CRH system within the CNS is crucially important for orchestrating broader behavioral responses to stress.
And it sounds like CRH is acting alone. It's part of a much larger, more complex family of related molecules.
Yes, the complex CRH system also includes non-hypophysiotropic CRH neurons, those not directly targeting the pituitary, plus 3 additional CRH-like peptides, urocortin 1, 2, and 3. And there are at least 2 cognate GPCRs, or receptor types, CRH receptor 1, CRHR1, and CRH receptor 2, CRHR2. Each of these receptors has distinct and complex distributions throughout the CNS. CRH peptides signal by binding to CRHR1 and CRHR2, which then couple to the stimulatory G protein, Gs and adenyl cyclists, initiating intracellular signaling cascades. CRHR2 in particular has multiple splice variants, meaning different forms of the same protein exist. CRHR1 is predominant in the brain and anterior pituitary, making it the primary receptor for CRH-mediated ACTH release. Urocortin 1 is a potent agonist. It activates the receptor strongly for both CRHR1 and CRHR2, while urocortins 2 and 3 are specific agonists of CRHR2. So there's specificity there.
There's also this CRH binding protein, which sounds a bit counterintuitive. Why would you have something that binds up your main stress hormone?
Yeah, the functional significance of the CRH binding protein, or CRH-BP, is quite interesting. It doesn't actually bind to the CRH receptor itself, but it does inhibit CRH action, likely by modulating CRH effects at the cellular level, maybe sequestering it. It's present in corticotroph cells in the anterior pituitary and in CNS regions that synthesize CRH or receive CRH-containing innervation, like the BNST and the central nucleus of the amygdala, both critical for stress processing. Critically, it appears to help regulate the stress response itself. Its levels increase in the amygdala following various types of stress like restraint stress, predator stress, and food deprivation. This suggests it reduces the anxiety-promoting effects of CRH by decreasing CRHR1 activation. For instance, transgenic mice that lack the CRH-BP gene show increased anxiety, underscoring its role in dampening the overall stress response.
So CRH truly has impacts way beyond just regulating our basic stress hormones like cortisol.
Absolutely. CRH and the urocortin peptides have a wide range of biological activities in addition to their hypophysiotropic role in releasing ACTH. Essentially, these peptides profoundly influence behavioral responses to stress, defensive behaviors to visual threats, anxiety levels, mood, arousal, locomotion, reward pathways, energy expenditure, and feeding behavior. They can even increase sympathetic activation, that fight-or-flight response, independently of the HPA axis itself. In the periphery, CRH is generally pro-inflammatory and acts as a paracrine factor in the endometrium, playing roles in decidulation, implantation, and uterine vasodilation. Studies using knockout mice lacking CRH, CRHR1, or CRHR2 have shown that while CRH is indispensable for mounting the acute stress response, other ACTH secretagogues can't fully replace it. CRH-independent mechanisms might augment glucocorticoid secretion after prolonged stress. This highlights its essential, but also interconnected function within a broader stress response system.
How do different types of stress specifically affect the HPA axis and its activation pathways? Is it always the same response?
That's a great question. Significant physiological or psychological stressors commonly activate both the HPA axis and the sympathoadrenal axis, releasing adrenaline and noradrenaline. While the end result, increasing circulating glucocorticoids, is often similar across various stress paradigms, different brain regions are actually recruited depending on the specific type of stress. For example, systemic stressors like cytokine treatment during an infection primarily activate catecholaminergic neurons in the solitary tract nucleus and ventrolateral medulla in the brainstem. In stark contrast, neurogenic, emotional, or psychological stressors, like the anticipation of a foot shock, activate not only these brainstem neurons but also cell groups in the limbic forebrain and hypothalamus. What's more, blocking the ascending catecholaminergic fibers blocks the HPA activation caused by systemic stress, but not the activation caused by foot shock. This clearly demonstrates distinct neural pathways are engaged depending on the nature of the stressor.
Okay, next, let's turn our attention to growth hormone and its primary stimulator from the hypothalamus, growth hormone-releasing hormone, or GHRH.
Right. In humans, GHRH occurs in two principal molecular forms, GHRH144-NH2 and GHRH148H. Both are derived from a larger prohormone, a precursor molecule. The NH2 terminal end is essential for its biological activity, but the COHR terminal NH2 group isn't strictly necessary. It's rapidly inactivated in the circulation by an enzyme called type IV dipeptidyl peptidase, which accounts for most of the immunoreactive peptide detected in plasma. This means it has a very short lifespan in the blood, allowing for pulsatile control. GHRH also shows species differences in its sequence, though the core function is conserved. For example, homology with the human peptide ranges from 93% in pigs down to 67% in rats.
And what can you tell us about the GHRH receptor, the molecule that translates the signal into action within the pituitary?
The GHRH receptor is a G-protein coupled receptor, a GPCR. When activated, it elevates intracellular KMP, increases intracellular free calcium, triggers the release of preformed GH from storage vesicles, and stimulates both GH messenger RNA transcription and new GH synthesis. Importantly, nonsense mutations, which lead to prematurely truncated non-functional proteins in the human GHRH receptor gene, cause rare familial forms of GH deficiency. This really highlights that no other gene product can fully compensate for this specific receptor's function. Furthermore, the proper functioning of the GHRH receptor on savannatrophs, the cells that produce GH, might also depend on another protein called RECK, which is expressed in most somatotrophs. Deficiency in RECK leads to decreased GHRH receptor levels and reduced IGF-1, a key growth factor stimulated by GH.
How does GHRH actually affect the overall pattern of GH secretion in the body? Does it just cause a steady release?
No, not quite steady. Intravenous administration of GHRH to individuals with normal pituitaries causes a prompt, dose-related increase in serum GH that typically peaks after about 15 to 45 minutes, followed by a return to basal levels within 90 to 120 minutes. While a maximally stimulating dose is around 1 microgram per kilogram of body weight, the response varies considerably between individuals. This is due to the influence of endogenous co-secretagogues, other stimulation factors, and the inhibitory tone from somatostatin. Interestingly, unlike the significant desensitization or loss of responsiveness seen with continuous GnRH administration, pulsatile GH secretion and IGF-1 production are actually maintained by constant GHRH infusion in humans. This suggests that other factors, likely including somatostatin pulses, contribute significantly to GH's intrinsic diurnal rhythm, its daily fluctuation.
It also seems that GHRH has some effects beyond just direct pituitary control of growth hormone.
Yes, that's correct. It has been reported to stimulate food intake in rodents and sheep, although its physiological relevance to feeding in humans is currently unknown. Beyond its primary role in GH release, non-pituitary GHRH, found in other tissues, can also modulate cell proliferation and even promote skin wound healing, indicating broader biological actions we're still uncovering.
There's also this fascinating class of substances called GH secretagogues or GHSs that have a bit of an interesting history, right? They weren't initially sought out for GH release.
Indeed. In earlier studies focused on opioid control of GH secretion, researchers unexpectedly found that several peptide analogs of metenkephalin, such as GHRP6 and hexaneuralin, were actually potent GH secretagogues, GHSs. Later, non-peptidyl GHRP mimetics, like MK0677, were synthesized. These offered the advantage of better oral bioavailability, meaning they could be taken by mouth. A common feature of these compounds is their activation of phospholipase C and inositol 1004-5 trisphosphate signaling pathways inside the cell. This ultimately led to the identification of a novel G-protein-coupled receptor, the GHS receptor, now often called the ghrelin receptor. This receptor is highly selective for GHS ligands, completely unrelated in structure to the GHRH receptor, and is highly expressed not only in the anterior pituitary, but also in various brain areas, including GHRH neurons themselves. This suggests both direct effects on the pituitary and indirect effects via the hypothalamus on GH secretion.
And it seems ghrelin, the hunger hormone from the stomach, is a key natural GHS connecting the gut directly to growth regulation.
Ghrelin, yes. It's an acylated 28 amino acid peptide produced primarily in the stomach, and it's another crucial natural ligand for the GHS receptor. Its O-N-octanoylation at SIR3, that unique and critical post-translational modification where an octanoic acid group is attached, is absolutely essential for its biological activity. Makes it quite unique. Ghrelin receptors are highly expressed on somatotrophs in the anterior pituitary and also in the hypothalamus, particularly in the arcuate nucleus, indicating both direct and indirect effects on GH secretion. When acylated, ghrelin stimulates GHRH release from hypothalamic explants and increases the firing rate of GHRH neurons. When continuously administered, it amplifies GH pulsatility. Its effect to increase GH seems unaffected by somatostatin's inhibitory actions, and in sheep, ghrelin even prevents somatostatin release into the portal system, further enhancing GH release. There's also clear evidence of a gastrohypophyseal feedback loop, as ghrelin levels fall in response to rising GH levels, completing the regulatory cycle.
What about insulin's surprising role in GH regulation? We typically think of insulin and glucose being tightly linked, but how does it affect growth hormone?
It's an intriguing connection that adds another layer. Somatotrophs, the GH-producing cells, actually express insulin receptors, and these might be required to maintain the sensitivity of the GHRH receptor. Intriguingly, selective knockdown of the insulin gene, specifically in the PVH, the paraventricular nucleus of mice, is associated with decreased serum GH levels. Furthermore, chronic restraint stress, which we know increases CRH in hypophysiotropic neurons, also reduces INSOS2 mRNA, the messenger RNA, for a specific insulin gene in the PVN. This leads to decreased GH mRNA and serum GH levels, ultimately impeding growth in young mice. This growth impairment can actually be prevented by overexpressing INS2 just in the PVN. The hypothesis is that decreased PVH insulin levels under stress contribute to growth retardation by suppressing pituitary GH production. It adds another layer of complexity connecting stress, metabolism, and growth.
So how do GHRH and somatostatin, these two key players with opposing actions, work together to create those distinctive GH pulses we see throughout the day?
Right, it's a delicate dance. GH secretion is primarily regulated by these two major hypothalamic peptides. GHRH stimulates and somatostatin inhibits. They function in concert to produce the rhythmic pattern of about 10 to 20 pulses of GH you experience over a 24-hour period. GHRH neurons are located mainly in the arcuate or infundibular nucleus of the hypothalamus, while somatostatin neurons involved in GH regulation are predominantly in the hypothalamic paraventricular nucleus. It's generally presumed that GH pulses are mainly driven by the withdrawal of somatostatin's inhibition, allowing GH release, while GHRH determines the amplitude or the size of these pulses. An additional mechanism for GH pulse generation might involve ultradian changes occurring more frequently than daily in synaptic transmission onto GHRH neurons, where increased excitatory input develops during periods of low circulating GH. But it's a very finely tuned system, as other circulating hormones and modulatory peptides at both the pituitary and hypothalamic levels further sculpt the final GH secretion pattern.
This entire system is clearly under exceptionally complex neural control. It sounds like a vast interconnected web reaching beyond the hypothalamus.
Indeed it is. Multiple extrahypothalamic brain regions, those outside the immediate hypothalamus, provide efferent connections. They send signals to the hypothalamus, actively regulating GHRH and somatostatin neuronal activity. Somatosensory and affective or emotional information is integrated and filtered through the amygdaloid complex. The basolateral amygdala, for instance, provides an excitatory input, while the central extended amygdala provides inhibitory input. Many intrinsic neurons within the hypothalamus itself also release GABA, often co-releasing it with a peptide neurotransmitter. Cholinergic fibers, mostly from hypothalamic interneurons, densely innervate the median eminence. Similarly, dopaminergic and histaminergic neurons originate locally within the hypothalamus and the arcuate and tuberomammillary nuclei, respectively, all contributing to this intricate regulation.
What about ascending pathways, the signals coming up from lower brain regions like the brainstem?
Two important ascending pathways reaching the medial basal hypothalamus critically regulate GH secretion. These are serotonergic neurons originating from the raphing nuclei in the brainstem, and adrenergic neurons coming from the nucleus of the tractus solitarius, NTS, and the ventrolateral nucleus of the medulla. Many other neuropeptides also modulate GH secretion, notably galanin, which stimulates GH in humans via a GHRH-dependent mechanism and also by facilitating catecholamine release presynaptically. Opioid peptides also stimulate GH release, likely by disinhibiting GHRH neurons, basically removing an inhibitory brake, with enkephalin specifically mediating this action.
It's interesting how GH levels change so dramatically throughout life. What happens during puberty, and what about the somatopause, which sounds, well, pretty definitive?
Puberty certainly brings significant changes, yeah. You see increased GHRH release in action, and consequently IGF-1 levels increase notably. However, in young adulthood, surprisingly, daily GH secretion tends to return towards prepubertal levels, despite continued high levels of gonadal steroids. This might be partly due to increased weight gain typically seen in early adulthood. The somatopause is indeed a striking term. It's defined by an exponential decline in the GH secretory rate, with a half-life estimated at about 7 years, starting as early as the third decade of life. This decline is generally more pronounced in men until women go through menopause, after which the decline rates might become more similar.
How do external signals in the body's metabolic state precisely fit into this complex regulation of GH?
Peripheral signals, especially those related to energy intake and metabolism, provide absolutely crucial cues between your peripheral tissues and the hypothalamic centers regulating GH. And this is an area where species-specific regulatory responses are particularly prominent, making direct extrapolations from, say, rodent models to human GH regulation sometimes unreliable. For instance, fasting, protein energy malnutrition, and hypoglycemia all stimulate GH secretion. This is likely due to decreased IGF-1 feedback, increased GHRH drive, decreased somatostatin inhibition, and reduced insulin levels. In contrast, postprandial hyperglycemia, high blood sugar after a meal glucose infusion, elevated free fatty acids, and obesity generally inhibit GH secretion. This inhibition is often associated with increased insulin, which can directly inhibit somatotrophs via their insulin receptors. Leptin's role is complex. It prevents the fasting-induced fall in GH in rodents, but doesn't seem to do the same in fasting humans, highlighting those species differences again. Steroid hormones also have complex actions. Acute glucocorticoids can initially increase GH, while high doses or long-term administration tend to reduce it. CRH neurons also inhibit GH, and physiological levels of thyroid hormones are necessary to maintain normal GH secretion and promote GH gene expression. It's a real integration of metabolic, hormonal, and stress signals.
Somatostatin really sounds like a universal inhibitor, given all its broad effects across so many systems.
It truly is. It was actually unexpectedly discovered during efforts to isolate GHRH. Instead, researchers identified this factor that potently inhibited GH release from the pituitary. Originally called somatostatin 14, SST14, because it's a 13 amino acid peptide, a second longer form, SST28, was later identified. Both are derived from a common prohormone precursor. SST14 is predominant in the brain, including the hypothalamus, while SST28 is found in major quantities in the gastrointestinal tract.
So its name, somatostatin, which literally implies inhibiting growth hormone or soma, seems far too narrow for its true range of functions.
Exactly. The name somatostatin is indeed descriptively inadequate given its scope. Beyond its effect on growth hormone, it also inhibits TSH secretion from the pituitary and has widespread non-pituitary roles as a neurotransmitter or neuromodulator in both the central and peripheral nervous systems. It also functions as a key regulatory peptide in the gut and pancreas. In the gut, it acts as both a neurotransmitter and a paracrine secretion, influencing adjacent cells. It can even influence its own secretion through an autocrine function and modulates gut exocrine secretion as a lumum, meaning it acts within the lumen or cavity of an organ. Because of its wide distribution, its broad spectrum of regulatory effects, and its ancient evolutionary history, it can really be regarded as an archetypical pan-system modulator. It affects almost everything.
Is its genetic makeup highly conserved, similar across different species, given its ancient origins and broad roles?
Yes. The genes encoding somatostatin in humans and many other species show striking sequence homology, meaning they are very, very similar, even when compared to primitive fish like the anglerfish. Furthermore, the amino acid sequence of SST14 itself is identical in all vertebrates studied so far. This underscores its profound evolutionary conservation and critical biological importance across the animal kingdom.
Where specifically is somatostatin expressed in the brain beyond the areas controlling GH?
Apart from its well-known expression in neurons of the periventricular and arcuate hypothalamic nuclei involved in GH secretion, somatostatin is highly expressed in numerous other brain regions. These include the cortex, the lateral septum, the extended amygdala, the reticular nucleus of the thalamus, the hippocampus, and many brainstem nuclei. The molecular mechanisms for somatostatin gene transcription have been extensively studied, particularly in pancreatic islet cells, but in neurons, its expression seems to be strongly controlled by the transcription factor CREB.
Tell us more about the various somatostatin receptors, the SSTRs, and how they contribute to its diverse actions. How many are there?
There are 5 distinct somatostatin receptor subtypes, logically named SSTR1 through SSTR5. SSTR2 actually has 2 alternatively spliced forms, meaning minor structural variations exist. These subtypes are encoded by separate genes and are expressed in unique or sometimes overlapping distributions across multiple target organs. They also differ significantly in their coupling to second messenger signaling pathways inside the cell. All SSTR subtypes are coupled to pertussis toxin-sensitive G proteins and bind both SST14 and SST28 with high affinity, although SST28 shows a uniquely high affinity for SSTR5. SSTR1 and SSTR2 are the most abundant subtypes found in the brain and likely function as presynaptic autoreceptors, helping to regulate the release of somatostatin itself. All subtypes are expressed in the pituitary, with SSTR2 and SSTR5 being the most abundant on somatotropes, GH cells. They're also physiologically important in pancreatic islets for regulating insulin and glucagon secretion.
What are its broad effects on target tissues throughout the body, fitting with that pan-system modulator description?
Right. In the pituitary, somatostatin clearly inhibits the secretion of GH, TSH, and, under certain conditions, even PRL and ACTH. Beyond the pituitary, it exerts profound inhibitory effects on virtually all endocrine and exocrine secretions of the pancreas, the gut, and the gallbladder. It also inhibits salivary glands and sometimes parathyroid hormone and calcitonin secretion. Importantly, somatostatin very effectively blocks hormone release in many different types of endocrine-secreting tumors, including insulinomas, glucagenomas, VPOMas, carcinoid tumors, and some gastrinomas. This makes it incredibly valuable clinically for managing these conditions.
And on that note, are there significant clinical applications for somatostatin itself or its analogs? Can we use this knowledge therapeutically?
Absolutely. An extensive pharmaceutical development program has produced several somatostatin analogs, like octreotide, lanreotide, and pasireotide. These offer improved pharmacokinetics, meaning they last longer and are easier to administer, and also enhanced receptor subtype selectivity compared to native somatostatin. These are agonists, typically with high affinity for SSTR2 and SSTR5. A new generation of non-peptidyl agonists, like baltusitine, offer selective subnanomolar affinity, meaning they bind very, very tightly, to all 5 SSTR subtypes, and have shown promise in suppressing GH and IGF-1 in conditions like acromegaly. Furthermore, somatostatin analogs labeled with radioactive tracers are now widely used as external imaging agents for a huge range of disorders. This includes most gastroenteropancreatic neuroendocrine tumors, GP-NETs, pheochromocytomas, medullary thyroid carcinoma, many pituitary tumors, and even some non-endocrine tumors that happen to express SSTRs. Since activated T cells of the immune system also display SSTRs, inflammatory lesions like sarcoidosis and certain lymphomas can also be visualized using these tracers. They've become powerful diagnostic and therapeutic tools.
Okay, now let's turn to prolactin, PRL. It has a truly unique regulatory mechanism compared to almost all other interior pituitary hormones.
Yes, what's really fascinating here is that PRL secretion is primarily under tonic inhibitory control by the hypothalamus. This means that if you disrupt the connection between the hypothalamus and the pituitary, say, by surgically destroying the stalk median eminence or by transplanting the pituitary to an ectopic or abnormal site outside its usual location, you actually get a marked increase in PRL secretion. This is the complete opposite of what happens with GH, TSH, ACTH, and gonadotropins, where disrupting the connection would typically cause their levels to decrease. Prolactin is held in check normally.
And it seems dopamine is the key player in this constant suppression, acting as the main brake.
Yes, dopamine is unequivocally the principal physiological prolactin inhibiting factor, or PIS. It's present in the hypothalamic, hypophyseal portal vessel blood at concentrations sufficient to inhibit PRL release, and dopamine D2 receptors are specifically expressed on lactotrophs, the pituitary cells that produce prolactin. The importance of this is really emphasized by studies in mutant mice. Mice with a targeted disruption of the D2 receptor gene develop lactotroph hyperplasia, and overgrowth of these cells hyperprolactinemia, which is elevated prolactin in the blood, and eventually they develop lactotroph adenomas, benign tumors. This powerfully highlights dopamine's critical importance, not just in regulating hormone secretion, but also in controlling lactotroph proliferation or growth. The intrinsic dopamine neurons originating in the medial basal hypothalamus form the key regulatory population providing this conic inhibition.
How exactly do these D2 receptors on the lactotrophs work at a molecular level to inhibit prolactin release and cell growth?
The binding of dopamine or selective D2 agonists like bromocriptine to the D2 receptor initiates multiple coordinated effects on lactotroph function. The D2 receptor inhibits adenyl cyclase, thereby decreasing intracellular cyclic AMP, AMP levels. It also activates certain potassium channels, decreases voltage-activated calcium currents coming into the cell, and inhibits N-acetyl phosphate production. All these intracellular signaling events converge to lead to decreased free calcium concentrations within the cell and consequently inhibition of PRL secretory granulocytosis, the actual release of prolactin. The precise mechanism by which D2 receptor activation inhibits PRL gene transcription is still debated, but it likely involves inhibition of downstream signaling pathways like MAPK or protein kinase C, which in turn reduces the activity of H-family transcription factors crucial for PRL gene expression. Similarly, multiple pathways involving activation of ERK12 and inhibition of AKT protein kinase B seem to be implicated in the D2 receptor's anti-proliferative effects on lactotrophs, preventing them from overgrowing.
Are there also prolactin-releasing factors, or PRFs, that stimulate its release, balancing out dopamine's constant inhibition? Or is it all inhibition?
No, there are definitely stimulatory factors too. Several candidate prolactin-releasing factors exist. Arginine vasopressin, AVP, for instance, is released during stress and hypovolemic shock, conditions where PRL is also released, suggesting AVP might act as a PRF in these specific contexts. Peptide histidine isoleucine, PHI, and its human homolog peptide histidine methionine, PHM, are also considered strong candidate PRFs, potentially playing a role during stress. Serotonin, originating from the brainstem dorsal raphe, plays a major role, particularly during lactation. It seems to work by inhibiting dopamine secretion from the TII neurons and potentially activating PRFs via TRH neurons. Endogenous opiates like dynorphin and beta-endorphin may also contribute by inhibiting dopamine secretion during lactation. So it's a complex interplay of stimulatory and inhibitory signals, not just dopamine.
So to reiterate that key point, PRL is truly under constant hypothalamic suppression, which is unique among the anterior pituitary hormones.
That's the fundamental principle for PRL regulation. Yeah, it really distinguishes it. Secretory bursts of PRL are generally caused by the acute withdrawal of that dopamine inhibition or stimulation by PRFs, or often a combination of both. Local autocrine and paracrine regulators within the pituitary further modulate the responsiveness of individual lactotrophs to these signals. This system is heavily scrutinized clinically because PRL hypersecretion syndromes, like prolactinomas, are relatively common, and many neuroleptic drugs used to treat psychiatric conditions commonly enhance PRL release precisely because they block dopamine action at the D2 receptor on the pituitary.
Does PRL secretion exhibit a circadian rhythm, like so many other hormones we've discussed? Does it vary across the day?
Yes, it does. PRL is secreted in discrete pulses, superimposed on a basal level of secretion, and it exhibits a clear diurnal rhythm, with peak values typically occurring in the early morning hours, often associated with sleep. In humans, this appears to be a true circadian rhythm, maintained independently of the sleep rhythm itself, even in a constant environment, suggesting it's driven by an internal clock, likely the SCN. This nocturnal rise in PRL might have evolved specifically for milk maintenance during prolonged non-suckling periods at night, ensuring milk availability for offspring. Endogenous diurnal alterations in dopamine tone, which are themselves entrained by light via the SCN, are thought to be the major neuroendocrine mechanism driving this rhythm. VIP, or vasoactive intestinal peptide, released from the SCN, is also a potential co-regulator. perhaps by inhibiting the TIDA dopamine neurons or acting via a PRF pathway.
What happens during suckling? That's such a powerful physiological stimulus for prolactin release in lactating mammals.
Suckling is incredibly powerful. It dramatically reduces dopamine output from the TIDA neurons, which lifts the inhibition on lactotrophs and presumably sensitizes them to PRFs. The complex neural pathways involved in the suckling reflex travel from mechanoreceptors in the nipple up to the spinal cord, ascend through hypothalamic tracts, and eventually enter the hypothalamus. While the neurons regulating the oxytocin-dependent milk letdown reflex separate off at the PVH, they accompany the PRL regulatory pathway throughout much of this ascending journey. Several brainstem relay centers are activated during suckling, leading to a coordinated inhibition of PIF dopamine activity and the release of PRFs. Although, as I mentioned, a single undisputed suckling-induced PRF hasn't been definitively identified yet. What's more, suckling-induced prolactin secretion also coordinates other crucial physiological responses vital for successful lactation. This includes the development of maternal behavior, increased appetite or hyperphagia, and the inhibition of reproductive function, preventing ovulation during early lactation. Prolactin binding to its receptors in the medial preoptic area, EPO, is critical for displaying maternal behavior. Blocking these receptors disrupts things like pup retrieval. The hyperphagia is explained by increased MPY and AGRP gene expression in the arcuate nucleus during suckling, mediated by brainstem afferents and a fall in circulating leptin. Activation of these arcuate MPY neurons may also contribute to the reproductive inhibition seen during suckling. It's a beautifully integrated system.
Okay, let's talk about gonadotropin-releasing hormone, or GnRH. This is really the master regulator of the entire reproductive axis, controlling puberty, fertility, everything.
Absolutely. GnRH is a 10-amino acid hypothalamic neuropeptide that centrally controls reproductive function in vertebrates. It's synthesized as part of a larger precursor molecule, pro-GnRH. All known forms of this decapeptide, a small peptide of 10 amino acids, have specific terminal residues. A pyroglutamate, pyroglu, at the NH2 terminus, and a glycinamide, gliamide, at the COO terminus. This structural conservation underscores their critical functional importance throughout evolution.
And it turns out there's actually more than one GnRH gene in mammals, adding another layer of complexity to the story.
That's right. 2 distinct genes encoding GnRH have been identified in mammals. GnRH1 encodes the form found predominantly in hypothalamic neurons, acting as the primary releasing factor for pituitary gonadotroph function controlling LH and FSH release. GnRH2, on the other hand, encodes the K-peptide that differs by 3 amino acids. It's found mainly in the midbrain, where it seems to act more as a neurotransmitter or neuromodulator rather than a pituitary releasing factor, suggesting distinct roles within the brain. Both GnRHI and GnRH2 are found across diverse species, from fish all the way to mammals. A third form, GnRH3, is present in teleost fish. Advanced imaging techniques are revealing that GnRH shells are even more diverse and widely distributed than previously thought, even appearing in the basal ganglia, although their function there isn't clear yet. GnRH is also found outside the brain in areas like mammary glands, ovaries, and the placenta, though its precise function in those locations is still not well understood.
How is the GnRH gene itself structured and regulated to ensure its very precise expression only in the right neurons and at the right times?
All GnRH genes share a basic 4-exon structure, meaning their coding sequences are split into 4 segments interrupted by non-coding introns. The nucleotide sequences encoding the decapeptide itself are highly homologous across species, indicating deep evolutionary conservation. Transcriptional regulation is highly complex, involving conserved promoter regions, DNA sequences that initiate transcription, and enhancer elements which boost transcription levels. Various transcription factors including OCT1, MSX, DLX, OTX2, VAS1, and BRN2 are critical for specifying its neuronal expression and controlling its developmental activation. The mouse GnRH gene even contains a specific kisspeptin response element, or KSRE, in its enhancer region. This functions by recruiting another transcription factor, OTX2, to binding sites within the KSRE, which then activates chromatin changes necessary for the KIS1 receptor signaling pathway to work. Other factors like TBX21, EGR1, ZB1, CABP, and even microRNAs like MIR200, MIR155, and MIR29 also contribute significantly to the intricate regulation of GnRH transcription. It's controlled by a whole network of factors.
And where specifically are these crucial GnRH neurons located within the brain? Are they clustered together?
Actually, no. Neurohypophysial GnRH neurons, the ones that project down to the median eminence to control the pituitary, are surprisingly widely distributed throughout the hypothalamus. You find them in the preoptic area, the infundibulum, arcuate nucleus, and the periventricular region, Interestingly, they're interspersed with non-neuroendocrine GnRH neurons, which project to other brain areas, including the cortex. These other GnRH neurons have been implicated in the control of sexual behavior in rodents, showing that GnRH has diverse functions beyond just hormone release.
GnRH neurons have a truly fascinating origin story, migrating from outside the brain itself during development.
Yes, what's truly fascinating here is that GnRH neuroendocrine neurons are a highly unusual neuronal population because they originate entirely outside the CNS. Specifically, they arise from the epithelial tissue of the nasal placode, which forms part of the developing nose. During embryonic development, these neurons undertake an incredible journey, migrating across the brain's surface into the hypothalamus. This migration is precisely guided by a temporary scaffolding of other neurons and glial cells, with neural cell adhesion molecules playing a critical role in directing their path towards their final destination.
And what happens if this crucial migration fails during development, if they don't reach the hypothalamus?
Failure of GnRH neuron migration leads to a condition called Kalman syndrome. In this syndrome, these critical neurons don't reach their final destination in the hypothalamus and thus fail to properly stimulate pituitary gonadotropin secretion. As a result, patients with Kalman syndrome do not enter puberty spontaneously. The X-linked form results from a deficiency of the KAL1 gene, which encodes a protein called anosmin-1, crucial for migration. These patients often present with other congenital midline defects, including anosmia, a complete loss of the sense of smell due to hypoplasia or underdeveloped growth of the olfactory bulb and tracts. Autosomal dominant forms are linked to mutations in other genes involved in migration or development, like FGFR1, FGF8, and PROCO2, PROKR2, among others. Interestingly, anti-Mullerian hormone, AMH, is also expressed by embryonic GnRH cells during their migration and acts as a potent stimulator of GnRH cell motility, helping to guide them. Crucially, exogenous GnRH, administered externally as a therapy, effectively treats this form of hypothalamic hypogonadism, demonstrating the hormone's direct utility.
Okay, once GnRH actually reaches the pituitary gonadotroph cells, how does it translate its signal into the release of LH and FSH?
GnRH binds to a specific 7-transmembrane domain G-protein-coupled receptor, a GPCR, located on the surface of pituitary gonadotrophs. This binding event then stimulates both the synthesis and the secretion of both LH and FSH. Physiologically, the number of GnRH receptors on these cells varies and typically correlates directly with the pituitary's capacity to secrete gonadotropins. The levels of receptor message, the mRNA, are carefully regulated by steroid hormones like estradiol and progesterone, and also by intracellular second messengers like calcium and protein kinase C, highlighting multiple layers of dynamic control over pituitary responsiveness.
And what's this interesting phenomenon called self-priming that happens with GnRH? How does it prime itself?
Right, here's where it gets really interesting and physiologically important. When GnRH stimulation to the pituitary declines, as seen naturally in states like lactation, undernutrition, or seasonal reproductive quiescence, the number of GnRH receptors on gonadotrophs drops dramatically. They become less sensitive. However, subsequent exposure to GnRH pulses can powerfully restore receptor number through a calcium-dependent mechanism that specifically requires new protein synthesis. This effect, where GnRH actually induces its own receptor, is called upregulation or self-priming. Only certain physiological frequencies of pulsatile GnRH can effectively augment GnRH receptor production, and these optimal frequencies differ among species. This self-priming effect plays a crucial role in generating the massive mid-cycle gonadotropin surge that triggers ovulation in spontaneously ovulating species like humans. Before the surge, an increased GnRH pulse frequency, combined with the sensitization of pituitary gonadotrophs by rising estradiol levels, makes the pituitary exquisitely sensitive to GnRH. This leads to that huge LH output needed for the follicle to rupture and release the egg.
The pulsatile nature of GnRH secretion truly seems incredibly important for proper reproductive function. Not just the presence of GnRH, but the pattern.
It's absolutely critical. The ensemble of GnRH neurons in the hypothalamus doesn't fire constantly. It fires in a coordinated, repetitive, episodic manner. This produces distinct pulses of GnRH that are released into the portal bloodstream, bathing the pituitary. This pulsatile stimulation then leads to distinct pulses of LH and FSH appearing in the peripheral circulation, often in a very precise 1.1 ratio with the GnRH pulses. Because sampling portal blood is invasive and impractical in humans, we typically indirectly assess GnRH secretion by monitoring the pulsatile LH secretion in peripheral venous blood samples taken frequently. Now, while a single GnRH pulse stimulates both LH and FSH release, and chronic pulsatile exposure supports the synthesis of both, Divergent patterns of LH and FSH secretion can occur, depending critically on the frequency of the DNRH pulses.
So how precisely does GnRH pulse frequency influence the balance and secretion of LH versus FSH?
Experimental studies where endogenous GnRH is blocked and replaced with exogenous pulsatile GnRH delivery clearly show that different frequencies lead to different LH to FSH ratios being secreted. For example, a slower pulse frequency, say 1 pulse every 3 hours, can lead to decreased LH secretion but actually increased FSH secretion, effectively elevating the FSH-LH ratio. In the normal human menstrual cycle, LH pulse frequency naturally increases during the follicular phase, reflecting increased GnRH pulsatility driving follicle growth, and then slows down significantly during the luteal phase after ovulation. Abnormally low GnRH pulsatility is associated with conditions like delayed puberty, hypothalamic amenorrhea, often seen in athletes or due to stress low weight, and obesity-related hypogonadism. Conversely, inappropriately high GnRH pulsatility is linked to conditions like precocious puberty and polycystic ovarian syndrome, PCOS. This raises an important question, especially for mastering this topic like the professionals. What about the quality or bioactivity of the hormones released? GnRH pulse frequency not only influences the amount and ratio of hormones released, but also modulates the structural makeup, specifically the glycosylation of the gonadotropins. LH and FSH share a common alpha subunit, but have unique beta subunits that confer specificity. The extent of glycosylation, particularly with sialic acid residues, is crucial for their physiological function. More sialic acid means a longer half-life in circulation, protecting them from rapid liver degradation. Less sialic acid leads to more potent but shorter-lived effects at their target receptors. Both the GnRH stimulation rate and feedback from ovarian hormones regulate this glycosylation process. For instance, slower GnRH frequencies during the follicular development phase are associated with greater FSH glycosylation, providing sustained support to the growing follicles. In contrast, the faster frequencies occurring just before the mid-cycle surge lead to lesser FSH glycosylation, yielding a more potent but shorter-lasting form of FSH, perfectly suited for triggering ovulation.
Okay, what are the main neural inputs to the GnRH neurons themselves? It sounds like they must receive a lot of different signals to integrate.
GnRH neurons indeed receive inputs from a wide, complex array of neurotransmitters, creating an intricate regulatory network. These include norepinephrine, dopamine, serotonin, acetylcholine, nitric oxide, GABA, which is generally inhibitory, glutamate, generally excitatory, kisspeptin, endogenous opiate peptides, inhibitory, NPY, and galanin. Broadly speaking, glutamate and norepinephrine provide stimulatory drive, pushing GnRH release, while GABA and endogenous opioid peptides provide substantial inhibitory drive, acting like brakes. However, among all these diverse inputs, the kisspeptin neuronal network has emerged as particularly essential, perhaps paramount, for directly regulating GnRH neurons, acting as a critical gatekeeper for reproductive function.
Kisspeptin really seems to be the key then, almost like a master switch for puberty and ongoing reproductive function.
It truly does seem that way. Kisspeptin, which is an arphamide-like peptide, is the natural ligand, the molecule that binds to and activates the G-protein coupled receptor, GPR54, now often called KISS1R. The critical importance of this system is dramatically underscored by genetic evidence in humans. Inactivating mutations of the KISS1 gene, encoding kisspeptin, or the KISS1 gene, encoding the receptor, result in hypogonadotropic hypogonadism, meaning a failure to go through puberty due to lack of gonadotropin stimulation. Conversely, activating mutations in KISS1 cause precocious puberty, an abnormally early onset. These crucial kisspeptin neurons are primarily found in two discrete areas in mammals. In humans and primates, they are in the preoptic area and the infundibular nucleus, equivalent to the arcuate nucleus. In rodents and ruminants, they are in the anteroventral periventricular nucleus, AVPV, rostral periventricular area of the third ventricle, RP3V, and the arcuate nucleus, with the largest population consistently residing in the arcuate and fundibular nucleus. Now, what's fascinating here is that while GnRH neurons do exhibit some intrinsic rhythmic electrical activity when studied in culture, this intrinsic rhythm doesn't correlate well with the actual in vivo GnRH pulses observed in a living organism. So the concept of the pulse generator, the mechanism that actually creates the pulsatile release pattern, has largely been ascribed to these kisspeptin neurons, particularly those in the arcuate and fundibular nucleus. These kisspeptin neurons express estrogen and progesterone receptors, making them sensitive to feedback from the ovaries, and they densely innervate GnRH neuronal cell bodies, and also their axon terminals, or dendrons, down in the median eminence. GnRH neurons, in turn, express the kisspeptin receptor, kisspeptin, making them responsive to this critical kisspeptin signal. Ultimately, pulsatile GnRH secretion clearly depends on pulsatile kisspeptin secretion. Interestingly, continuous kisspeptin administration can actually suppress LH secretion over time, highlighting the absolute importance of the pulsatile pattern for sustained function.
So how exactly do these specific kisspeptin neurons in the arcuate and fundibular nucleus generate those critical pulses? What's the proposed mechanism?
The mechanism for generating pulsatile kisspeptin secretion, and thus GnRH pulses, is proposed to be mediated by at least two additional peptides that are co-expressed by these same kisspeptin neurons, operating with opposing actions in a sort of mini-circuit. These are neurokinin B, NKB, which acts via NK3 autoreceptors to stimulate kisspeptin secretion, and dynorphin, or possibly enkephalin in humans, which acts via opioid autoreceptors to inhibit kisspeptin secretion. This specific population of kisspeptin neurons is often referred to as KNDI, standing for kisspeptin, neurokinin B, and dynorphin. Again, human genetics provides strong evidence. Inactivating mutations in the genes for NKB or its receptor, NK3R, also result in hypogonadotrophic hypogonadism, just like mutations in kisspeptin Kes1R, confirming their essential role in the pulse generator. Synchronization of KNDI neuronal activity, creating those coordinated pulses across the network, might be achieved by interconnections among the KNDI neurons themselves and potentially through cellular communications with nearby astrocytes via gap junctions.
Does kisspeptin always act directly on the GnRH neurons themselves, or is there more nuance to where its action takes place?
That's a good point of clarification. While kisspeptin neurons do innervate both the GnRH cell bodies located further up in the hypothalamus and also their dendrins, those specialized dendrite-like processes that release hormones down in the median eminence, its primary action on GnRH secretion in primates appears to occur predominantly in the median eminence itself. Evidence suggests this activation of GnRH dendrins happens mainly by volume transmission, meaning the kisspeptin signal diffuses through the extracellular space to reach its targets, rather than requiring direct classical synaptic inputs onto the dendrins. Interestingly, NKB and dynorphin, although they are released by KNDI terminals in the median eminence alongside kisspeptin, seem to have no direct effect on the GnRH dendrins themselves, unlike their potent effects back on the KNDI neurons, autoregulation. This raises an important question, particularly for human physiology. While kisspeptin is clearly required for GnRH secretion, the strict pulsatility of kisspeptin itself might not be absolutely required in humans. Studies have shown the continuous infusion of kisspeptin 10 can still increase LH pulse frequency even in individuals who have mutations in the NKB receptor, which should disrupt the KND pulse generator. This suggests there might be other pulse generators operating upstream of, or parallel to, the KNDI neurons in humans that contribute to the overall pulsatile GnRH output.
And a newer peptide has recently been identified that also seems to play a role in this complex gonadal axis regulation. What can you tell us about phenixin?
Yes, phenixin 20-amide, or PNX, is a relatively novel peptide that is highly conserved across species, indicating its ancient and likely vital role. It also appears to regulate the gonadal axis, partly by sensitizing pituitary gonadotrophs to GnRH, making them more responsive, and potentially through direct effects on GnRH neurons as well. Intriguingly, PNX is synthesized in kisspeptin neurons, and it actually increases Kes1 mRNA levels, suggesting a potential positive feedback or autocrine loop. Moreover, kisspeptin neurons express the GPR173 receptor, for which PNX is a proposed ligand. This suggests PNX might activate kisspeptin neurons through an autocrine mechanism acting back on the cell that produced it, or through other intricate connections within the network. It adds yet another layer of complexity and potential regulation to this already rich system governing reproduction.
How do gonadal steroids like estrogen and testosterone produced by the ovaries and testes feed back into this hypothalamic pituitary system to regulate their own production?
Gonadal steroids, primarily estradiol and progesterone from the ovaries, and testosterone from the testes, provide absolutely crucial feedback signals that act at both the hypothalamus and the pituitary level. In primates, including humans, estradiol exerts considerable negative feedback directly at the pituitary, influencing the sensitivity and secretory capacity of the gonadotrophs. However, most of the negative feedback effects of progesterone and testosterone appear to occur predominantly at the hypothalamic level, likely influencing the activity of GnRH neurons, or their upstream regulators, like the KNDI neurons.
And estradiol also has that fascinating, powerful, positive feedback action that is absolutely essential for triggering ovulation in females.
Yes, here's where it gets really interesting and absolutely critical for reproduction in females. In addition to its predominantly negative feedback effects, estradiol, when it reaches sustained high levels, can exert a powerful positive feedback action at both the hypothalamus and the pituitary. This leads to a massive, rapid release of LH and FSH from the pituitary, known as the LHF-FSH surge. This surge occurs once each menstrual cycle, and is the precise physiological trigger for ovulation, the release of the egg from the dominant follicle. This switch to positive feedback is a direct response to the rising tide of estradiol produced by the maturing dominant follicle during the late follicular phase of the cycle. In women, elevated estradiol levels, typically sustained at around 300 to 500 picograms per milliliter for about 36 hours, reliably precede the gonadotropin surge.
So what exactly drives this powerful LH-FSH surge? Is it just the high estrogen, or is the hypothalamus doing something different?
The positive feedback actions of estradiol occur at both levels. At the hypothalamus, where it appears to increase GnRH secretion, both frequency and amplitude, creating a GnRH surge, and at the pituitary, where it significantly enhances the gonadotrop's responsiveness to that GnRH surge. Remember the self-priming effect. At the hypothalamus, a distinct population of kisspeptin neurons located in the preoptic area, specifically the AVPVRP3V in rodents, analogous regions in primates, are believed to function as the surge center responsible for inducing the ovulatory GnRH surge. This subpopulation of kisspeptin neurons is specifically activated in an estrogen-dependent manner immediately preceding the GnRH surge, and is postulated to play the key role in mediating estradiol's positive feedback effect on GnRH release. Lesions in areas near the medial preoptic area, for instance, can specifically block the estrogen-induced surge without affecting the normal negative feedback mechanisms. The precise molecular mechanisms mediating the switch from negative to positive feedback are still not fully understood, but they involve complex estrogen-induced changes in transcription factors, receptor expression, and local circuitry. It's proposed that two functionally independent neuronal circuits are involved: the GnRH pulse generator, the KNDI neurons in the arcuated fundibular nucleus, and the GnRH surge generator, these preoptic kisspeptin neurons. They seem to operate in parallel but independently. The pulse generator's frequency, for example, remains relatively constant even during the GnRH surge, indicating it's not directly responsible for generating the surge itself.
When does the reproductive axis actually wake up and begin its activity in human development? Is it just dormant until puberty?
Actually, no. Neuroendocrine stimulation of the reproductive axis initiates surprisingly early during fetal development, particularly in primates like humans during mid-gestation. There's a period of activity then, followed by relative quiescence during childhood. The well-known pubertal reawakening occurs in late childhood, typically marked initially by characteristic nighttime elevations in gonadotropin, especially LH, and gonadal steroid hormone levels during sleep. While the complete trigger mechanism for puberty isn't fully understood, significant progress has been made in identifying the central hypothalamic changes involved. At puberty, there seems to be a crucial decrease in transsynaptic inhibition, like from GABA, and a corresponding increase in stimulatory input, like from glutamate and kisspeptin, onto the GnRH neurons, effectively shifting the balance towards activation and pulsatile release.
Can you elaborate on what these specific inhibitory and stimulatory changes are thought to be?
Sure. One major inhibitory input that decreases is thought to come from GABAergic neurons. Studies in rhesus monkeys show that hypothalamic GABA levels and turnover decrease during early puberty, and experimentally blocking GABAergic input can prematurely activate the GnRH system in prepubertal animals. Conversely, a subset of glutamate receptors, specifically kainate receptors, seem to increase their activity around puberty, suggesting that the observed decrease in GABA tone might be, at least partly, due to increased excitatory glutamatergic transmission driving things forward. Administering NMDA, an activator of glutamate receptors, to prepubertal monkeys, for instance, can actually drive the reawakening of the reproductive axis. Increased stimulatory drive also likely comes from increases in norepinephrine and possibly NPY activity at puberty. Growth factors, acting perhaps through prostaglandin release from glial cells surrounding the GnRH neurons, might also stimulate GnRH neurons. Leptin, the hormone secreted by fat cells indicating energy stores, also plays a crucial permissive role. Neurons in the ventral premammillary nucleus, PMV, that project to kisspeptin neurons and express leptin receptors may mediate leptin's stimulatory effect on kisspeptin release. Re-expressing the leptin receptor just in the PMV of leptin receptor knockout mice can actually induce puberty. Neurotransmitters produced in these PMV neurons, like PACAP, glutamate, and neuronal nitric oxide, are also involved. For example, selective deletion of PACAP from PMV neurons leads to delayed puberty. Finally, epigenetic mechanisms, changes in gene expression without altering the DNA sequence itself, like methylation, are also proposed to help coordinate the multiple complex gene expression changes that accompany the onset of puberty. It's a multifactorial process.
How does stress, both physical and psychological, impact reproductive function? It feels like a very delicate balance that can be easily disrupted.
It is indeed a delicate balance, and stress can significantly impact it. Many forms of physical stress like fasting, intense exercise, temperature extremes, infection, pain, and injury, as well as psychological stresses such as social subordination or significant emotional distress, can profoundly suppress the activity of the reproductive axis. Brief stress might cause only acute temporary suppression of circulating gonadotropins and gonadal steroids, maybe leading to menstrual cycle disruption in females, though fertility might not be permanently impaired. However, prolonged periods of significant stress can lead to complete impairment of reproductive function, characterized by chronically low gonadotropin and steroid levels, a state sometimes called hypothalamic amenorrhea or functional hypothalamic hypogonadism. Stress appears to primarily exert its effects by decreasing the GnRH drive coming from the hypothalamus to the pituitary, as administering exogenous GnRH pulses can often reverse these suppressive effects, pointing to the hypothalamus, likely involving CRH and opioid pathways inhibiting GnRH-Gespeptin, as the key point of regulation.
And it seems metabolism, or energy balance, also plays a very direct and important role in reproductive control.
It absolutely does. Various metabolic fuels, including glucose and fatty acids, directly regulate reproductive axis function. Blocking their utilization, for example, can suppress gonadotropin secretion. Leptin, that crucial hormone from fat cells signaling energy sufficiency, also critically modulates the reproductive axis. Leptin-deficient mice are infertile, but remarkably, their fertility can be fully restored with leptin administration. Leptin receptors are found in key hypothalamic populations, particularly Gespeptin neurons, especially the KNDI neurons, influencing them through various mechanisms. These might include increased AMP-activated kinase, AMPK, phosphorylation during negative energy balance, signaling low energy, alterations in DNA methylation affecting gene expression, increased signaling by inhibitory peptides like NPY and AGRP, and reduced signaling by stimulatory peptides like alpha-MSH. Treatment with a combination of a neurokinin B agonist and a dynorphin antagonist targeting the KND system can even recover suppressed GnRH and LH secretion in some models of metabolic suppression. It appears that different neural circuits might mediate the effects of different types of stress, for example, metabolic versus psychological, on the GnRH system, adding another layer of specificity.
What about the connection between the hypothalamus and regulating sexual behavior itself beyond just the hormones?
The medial amygdala, part of the limbic system, is a critical node for processing sexually relevant stimuli, like pheromones, that profoundly influence mating behavior. A specific subset of Gespeptin neurons has been identified in the medial amygdala. These neurons are reciprocally connected to the accessory olfactory bulb, which processes pheromonal signals, and they also directly innervate GnRH neurons. This strongly suggests their involvement in coordinating pheromone-associated reproductive behavior with hormonal release. For example, exposing male mice to female urine activates neurons in the medial amygdala, and experimentally activating these medial amygdala Gespeptin neurons using chemogenetics leads to an increase in LH secretion, directly linking this specific circuit to reproductive function and potentially behavior.
Okay, beyond its massive role in hormone regulation, the hypothalamus is absolutely vital for other core body functions, like keeping our body temperature stable, right? Keeping us warm or cool.
That's absolutely right. The hypothalamus orchestrates a number of functions critical for normal homeostasis, including maintaining a stable core body temperature and also resetting it under specific physiological conditions, like inducing a fever during an infection. It has long been recognized that the brain plays an important role here, mediated by both warm-sensitive and cold-sensitive neurons, with the preoptic area, or POA, being the most important central thermoregulatory locus. Neurons in the POA serve as a major integrating center. They receive information from peripheral thermoreceptors in the skin, spinal cord, and deep abdominal structures. What's really neat is that some POA neurons even have the intrinsic ability to directly sense changes in local brain temperature, acting as their own internal thermometers.
So if the body gets too hot or too cold, what specific actions does the POA initiate to correct the temperature imbalance?
When the preoptic area warms up, it orchestrates responses to reduce heat production and increase heat loss. This includes triggering vasodilation, where blood vessels near the skin expand to radiate heat, inducing sweating, or panting in some animals, increasing respiration, inhibiting the activity of uncoupling protein 1, UCP1, in brown adipose tissue, BAT, which normally generates heat, and promoting behavioral responses, like seeking shade or a cooler area. Conversely, if the preoptic area cools down, it increases heat production and reduces heat loss. This is achieved by inducing vasoconstriction to conserve heat, initiating shivering, activating UCP1 in bat to generate heat metabolically, increasing overall intermediary metabolism, and promoting behaviors like increased locomotor activity to generate heat and increased food intake. Because warm-sensitive neurons significantly outnumber cold-sensitive ones in the POA, lesioning this region typically results in hyperthermia, an uncontrolled rise in body temperature. This leads us to ask, what are the precise central mechanisms downstream of the POA? The targets are primarily components of the sympathetic nervous system, including neurons in the dorsomedial hypothalamus, DMH, sympathetic premotor neurons in the rostral ventromedial medulla, RRPA, and preganglionic autonomic neurons in the spinal cord. Several distinct POA regions work in synchrony to relay this thermal information. For example, the median preoptic nucleus, MNPO, responding to skin cooling signals, initiates shivering, heat generation from bait, and vasoconstriction, likely by releasing the inhibitory transmitter GABA onto neurons in the medial preoptic nucleus, VPO. Lesioning the DMH or the RRPA effectively blocks shivering and bat activation.
So it's really specific neuronal populations within this POA area that are responsible for driving these very precise thermal responses.
Precisely. Modern molecular profiling techniques have identified specific populations. For example, GABAergic neurons in the ventromedial preoptic nucleus, VMPO, that co-produce the peptides PCAP and BDNF. When these specific neurons are activated, using optogenetics controlling them with light, it leads to a rapid decline in core body temperature. This happens via vasodilation and suppression of B heat production, as well as inducing behavioral cooling responses. Similarly, activating GABAergic neurons in another area, the ventrolateral preoptic nucleus, VLPO, also triggers a rapid temperature reduction and decreased physical activity. Conversely, inhibiting these VLPO neurons causes core temperature to rise. It's incredibly specific.
Is there also a direct link between the preoptic area's role in thermoregulation and our daily sleep cycles? Do temperature and sleep influence each other via the POA?
Absolutely, there's a strong link. The preoptic area also plays a significant role in mediating the circadian control of temperature regulation that's closely associated with sleep. In humans, our core body temperature naturally decreases during sleep, which is presumed to be an energy conservation strategy. Interestingly, warming the body before sleep, such as by taking a warm bath, can actually induce longer and deeper sleep episodes. This effect seems to be mediated through sensory afferents signaling warmth to preoptic neurons. It's proposed that temperature-activated glutamate neurons in the NPOMPO region innervate galanin GABA neurons in the VLPO, which is also a key sleep-promoting center. And this connection helps induce both NREM sleep and the accompanying hypothermia or temperature drop. Supporting this, ablation or removal of these galanin neurons elevates body temperature and prevents sleep rebound after deprivation, while artificially activating them induces hypothermia. Additionally, another hypothalamic region, the ventromedial nucleus, VMN, contributes to thermoregulation via the sympathetic nervous system's action on BAT, promoting the browning of white adipose tissue, making it more metabolically active in response to chronic cold exposure. Peripheral signals like thyroid hormone, GLP-1, estradiol, and leptin can act on the VMN to promote BT thermogenesis, partly by inhibiting AMPK activity. This has led to considerable interest in developing AMPK inhibitors as potential obesity treatments.
Sleep is such a critical, fundamental part of our lives, and it's clear the hypothalamus is deeply involved in orchestrating the whole sleep-wake cycle.
Sleep is indeed a fundamental biological process. It's a natural, reversible state of altered consciousness, typically divided into NREM, or non-rapid eye movement sleep, which includes the deepest stages characterized by slow brain waves, and REM sleep, characterized by rapid eye movements, vivid dreams, and increased autonomic activity. The neural organization controlling sleep and wakefulness relies on multiple interconnected brain regions. The primary sleep-promoting centers are indeed located in the ventrolateral preoptic nucleus, VLPO, and the median preoptic nucleus, MNPO, of the hypothalamus. These areas contain inhibitory neurons that use GABA and the peptide galanin as neurotransmitters. Other neurons that promote sleep include MCH, melanin-concentrating hormone, and GHRH neurons, also located within the hypothalamus.
And what are the key systems or centers that keep us awake, acting as the counterbalance to those sleep centers?
The wakefulness-promoting centers are actually more numerous and widely distributed, forming a robust arousal network. They include glutamatergic neurons in the brainstem reticular formation, and GABAergic, cholinergic, and glutamatergic neurons in the basal forebrain. Other absolutely key wake-promoting nuclei include the noradrenergic neurons in the locus coeruleus, serotonergic neurons in the dorsal raphe nucleus, dopaminergic neurons in the ventral periaqueductal gray, histaminergic neurons in the tuberomammillary nucleus, TMN, located right within the hypothalamus, and the crucial orexin, also known as hypocretin, glutamatergic neurons located in the lateral hypothalamic area. The reason for having such a large number of distinct wake-promoting cell groups remains a bit uncertain, but perhaps it serves a redundant purpose, ensuring we can maintain vigilance when necessary.
So how are these incredibly complex circuits for sleep and wakefulness interconnected and coordinated? How does the brain flip between states?
Wake-promoting pathways ascend from the brainstem into two major divisions. There's a dorsal pathway that projects primarily to the thalamus, which then relays signals to the cortex, and a ventral pathway that innervates the hypothalamus, the basal forebrain, and the cortex more directly. Both pathways are absolutely required for maintaining normal wakefulness. Within the hypothalamus, the TMN histaminergic neurons excite neurons throughout the cortex and thalamus, and also actively suppress activity in the sleep-promoting VLPO, thus promoting wakefulness. The lateral hypothalamic orexin neurons are also critical. They excite the TMN and project widely to other arousal centers, strongly promoting wakefulness and stabilizing the state. NREM sleep is primarily driven by the inhibitory neurons in the VLPO and MNPO, which actively inhibit these various wakefulness centers. The hypothalamic VLPO also contributes to initiating REM sleep by innervating specific brainstem regions that promote REM sleep features. This naturally leads us to ask, can we override this fundamental sleep-wake cycle when survival depends on it? And the answer is yes. The brain is equipped with mechanisms to override normal sleep pressure when necessary. Food deprivation, for instance, leads to increased wakefulness and locomotor activity, presumably to facilitate foraging. Orexin-producing neurons in the lateral hypothalamus played a major role in this response. Animals with ablated orexin neurons failed to show this increased wakefulness during fasting. Orexin neurons act as a critical interface between wakefulness and energy homeostasis, maintaining arousal during periods of food unavailability or high motivation. They are inhibited by signals of energy abundance like glucose and leptin, and powerfully activated by signals of hunger like ghrelin. These neurons project widely throughout the CNS, exciting wakefulness centers and inhibiting the sleep-promoting VLPO. The critical importance of orexin in promoting arousal is highlighted by the fact that loss of these neurons causes narcolepsy. This has led to the development of orexin receptor antagonists for treating insomnia and agonists for treating narcolepsy, some of which are currently in clinical trials.
So, we've seen how incredibly complex and finely tuned this hypothalamic pituitary system is. What happens when the system goes wrong, when things get out of balance?
Right. Disorders of the hypothalamic pituitary unit can arise from lesions or damage occurring at several different levels along the axis. Defects can stem from destruction of the pituitary gland itself, caused perhaps by a tumor, an infarct, tissue death due to lack of blood flow, inflammation, autoimmune disease, or even hereditary deficiencies like mutations in the PROP1 gene, which can lead to combined deficiencies in GH, prolactin, and TSH. Selective loss of specific hormone receptors just within the pituitary can also cause problems, like loss of thyroid hormone receptors leading to high TSH and thyrotoxicosis, or loss of GnRH receptors causing hypogonadotropic hypogonadism. Furthermore, disruption of that critical contact zone between the pituitary stalk and the median eminence, damage to the stalk itself, or damage to the nerve terminals of the tuberoinfundibular system, releasing factors into the portal blood can cause issues. This might be seen after surgical stalk section, with tumors compressing or involving the stalk, or in inflammatory disorders like sarcoidosis or autoimmune neurohypophysitis. At higher levels within the hypothalamus or its inputs, the loss of tonic inhibitory and excitatory inputs can manifest in various ways, such as an absence of normal circadian rhythms or the development of precocious puberty. Physical stress, the release of cytokines from inflammatory cells during illness, exposure to toxins, and even reflex inputs from peripheral homeostatic monitors can all impinge on and disrupt this system. And importantly, even severe emotional stress and psychological disorders can significantly activate the pituitary adrenal stress response, HPA axis, and suppress gonadotropin secretion, leading to conditions like psychogenic amenorrhea or inhibit GH secretion, contributing to psychosocial dwarfism.
What precisely is pituitary isolation syndrome? What happens when the pituitary is cut off from the hypothalamus?
This syndrome occurs when there are destructive lesions of the pituitary stalk, effectively isolating the pituitary from direct hypothalamic control. This can happen due to head injury, surgical transection, tumors, granulomas, or infiltrative diseases affecting the stalk. A large percentage of patients, depending on the level of the stalk damage, develop central diabetes insipidus, DI, where the body can't properly conserve water due to lack of AVP release from the posterior pituitary. A high cut, close to the hypothalamus, almost always produces permanent DI. With acute stalk section, less than half of patients experience the classic triphasic response. This involves an initial phase of polyuria, excessive urination, due to AVP neuron shock, followed by a brief period of normal water control or even SIADH, syndrome of inappropriate ADH secretion, as stored AVP leaks from dying terminals, and then finally the onset of permanent AVP deficiency over 1 to 10 days as the neurons degenerate. Interestingly, some recovery of water balance can occur even after months or years due to potential sprouting and regeneration of nerve terminals lower down. Non-destructive injury can also lead to transient DI or delayed SIADH.
And beyond diabetes insipidus affecting water balance, what other endocrine changes are typically observed after stalk damage isolates the anterior pituitary?
While menstrual cycles often cease after stalk section due to loss of pulsal GnRH stimulation, although some basal levels of gonadotropins might still be detectable. Plasma glucocorticoid levels and urinary cortisol excretion decline, but often more slowly than after a complete hypophysectomy, removal of the pituitary, as some basal ACTH secretion might persist, or there might be some CRH reaching via systemic circulation. A transient increase in cortisol secretion can initially occur due to the release of preformed ACTH stores from the damaged pituitary. The fall in GH secretion is generally considered the most sensitive indicator of stalk damage, but the insidious nature of this gradual decline in adults who have suffered traumatic brain injuries means it can unfortunately be overlooked. Causes of acquired central DI beyond trauma and surgery include granulomas like tuberculosis or sarcoidosis, tumors like craniopharyngiomas or metastases, autoimmune hypophysitis, Langerhans cell histiocytosis, and Erdheim-Chester disease. Congenital central DI can also be hereditary, often linked to mutations in the AVP gene itself.
What happens when specific hypothalamic hormones themselves are deficient, rather than a problem with the pituitary gland or the stalk connecting them?
Right. Selective pituitary hormone failure can indeed be caused by a deficiency of one or more specific hypothalamic releasing or inhibiting hormones, or sometimes by defects in the pituitary cell types themselves. Isolated GnRH deficiency is actually the most common specific hypophysiotropic hormone deficiency. We talked about Kallman syndrome. The X-linked form often involves both gonadotropin deficiency and hyposmia or anosmia, impaired or absent sense of smell. An MRI in these cases may show hereditary agenesis or failure of development of the olfactory lobe, reflecting that shared developmental origin and migration path of GnRH and olfactory neurons. Other midline brain malformations, like absence of the septum pellucidum and septo-optic dysplasia, can also cause hypogonadotropic hypogonadism.
What about hypothalamic amenorrhea? That seems like a relatively common condition affecting women.
Hypothalamic amenorrhea is a diagnosis often made in young, non-pregnant women who stop menstruating but have no identifiable structural abnormalities in their brain, pituitary, or ovaries. It's often considered functional, meaning it's due to factors like psychological stress, including conditions like pseudocyesis or false pregnancy, anorexia nervosa, excessive exercise, or sometimes hyperprolactinemic states. High prolactin can suppress GnRH. Psychogenic amenorrhea, linked to psychological stress, is the most common cause of secondary amenorrhea, cessation of periods after they've started. It can sometimes be temporary and related to relatively minor psychic stress. Anorexia nervosa, characterized by self-induced starvation and low body weight, very commonly leads to hypothalamic amenorrhea due to a profound loss of normal LH pulsatility. This is likely secondary to suppressed leptin levels signaling energy deficit, which then impacts kisspeptin GnRH signaling. Interestingly, while circulating kisspeptin levels themselves may not always differ significantly, serum levels of phenixin, that other regulatory peptide, are often lower in women with anorexia nervosa. In adult men, severe emotional stress or very strenuous exercise can also induce hypogonadism, although the symptoms might be more subtle than amenorrhea in women. Chronic opiate administration, often used for pain management, can also lead to hypogonadotropic hypogonadism by suppressing the GnRH pulse generator.
And what about specific deficiencies in TRH and GHRH, leading to problems with thyroid function and growth?
TRH deficiency is a rare cause of hypothalamic hypothyroidism. Molecular genetic analyses have revealed some rare autosomal recessive mutations in both the TRH gene itself and the TRH receptor gene. GHRH deficiency appears to be the principal cause of GH deficiency in many children presenting with idiopathic dwarfism, short stature of unknown cause. This is sometimes associated with abnormal EEGs or a history of birth trauma, suggesting potential subtle hypothalamic injury. While children with isolated GH deficiency often have normal-sized pituitary glands on MRI, those with combined pituitary hormone deficiencies, lacking multiple hormones, are more likely to show moderate to severe anterior pituitary hypoplasia, underdevelopment, or even pituitary stalk agenesis, absence of the stalk. As we discussed, the fall in GH secretion is the most sensitive indicator of stalk damage. A substantial GH response to provocative testing with a GHS like hexerolin typically only occurs if the vascular portal system in the stalk is at least partially intact. This leads us to consider adrenal insufficiency. Is it often caused by CRH deficiency?
Actually, adrenal insufficiency is rarely caused by primary CRH deficiency, although a genetic linkage to the CRH gene locus has been suggested in a few families with isolated ACTH deficiency. Mutations in the TBX19 gene, which encodes a crucial pituitary transcription factor called TPIT, are associated with most cases of isolated ACTH deficiency presenting in neonates, pointing to a pituitary developmental defect rather than a hypothalamic one in most cases.
Growth failure, particularly in children, can also be a significant sign of underlying hypothalamic problems, not just pituitary ones.
That's right. The loss of the normal nocturnal increase in GH secretion during sleep and deficient GH responses to standard provocative stimuli are early and highly sensitive indicators of hypothalamic dysfunction affecting the GHRH somatostatin balance. Anatomic malformations of midline cerebral structures, such as optic nerve dysplasia, underdevelopment of the optic nerve, are frequently associated with abnormal GH secretion, suggesting a shared developmental vulnerability. Certain complex genetic syndromes, including Prader-Willi syndrome, also commonly involve reduced GH secretory capacity, likely due to hypothalamic dysfunction.
What exactly is psychosocial dwarfism, and how is it linked specifically to hypothalamic function being disrupted by environment?
Severe infant neglect or abuse can lead to profound growth impairment and a failure to thrive, a condition sometimes termed maternal deprivation syndrome. In older children experiencing severe emotional disturbance or deprivation, a condition known as psychosocial dwarfism can develop. This is characterized by abnormal circadian rhythms, particularly blunted sleep-related GH release, and often deficient GH responses to provocative testing. Sometimes deficiencies in ACTH and gonadotropins can accompany this. A variant called hyperphagic short stature has also been identified, where children are short but have increased appetite. What's truly remarkable and highlights the plasticity of the system is that these disorders can often be significantly reversed simply by placing the child in a supportive, nurturing environment. This can lead to rapid catch-up growth and normalization of neuroendocrine responses. The exact pathogenesis of the altered GH secretion in these children remains unknown, but it clearly involves hypothalamic dysregulation triggered by severe environmental stress.
And what about neuroregulatory GH deficiency? It sounds like there's still some uncertainty surrounding that specific diagnosis.
Yes, this term refers to a group of children who exhibit low growth rates and often have low levels of IGF-1, the downstream mediator of GH action, but who surprisingly show a normal GH secretory reserve when given standard provocative tests. They often have demonstrably abnormal spontaneous GH secretion patterns when measured over 24 hours, with reduced pulse frequency or amplitude. Despite having normal stimulated GH levels, they may grow normally only when treated with exogenous GH. There's considerable uncertainty about the precise diagnostic criteria for this condition, and often a poor correlation between the observed spontaneous GH secretion patterns and the actual benefit from GH treatment. This suggests that underlying genetic defects affecting GH action or subtle hypothalamic dysregulation might be involved that aren't fully captured by current testing methods.
While too little of these crucial hormones is clearly problematic, too much can also lead to significant issues, these hypersecretion syndromes. Can they originate from the hypothalamus?
Yes, absolutely. Pituitary hypersecretion, meaning the pituitary releases too much of a hormone, can occasionally be caused directly by tumors or lesions within the hypothalamus that are overproducing, releasing hormones. For instance, GnRH secreting hamartomas, those benign tumor-like growths of normal tissue in an abnormal location, often near the hypothalamus, can cause central precocious puberty by acting as an ectopic GnRH pulse generator. Gangliocytomas, which are rare neuronal tumors often found in or near the hypothalamus and sometimes combined with pituitary adenomas, frequently lead to hormonal hypersecretion. Examples include CRH-secreting gangliocytomas causing Cushing syndrome due to excess ACTH, GHRH-secreting ones causing excess TSH, and very rarely TRH-secreting gangliocytomas causing thyroid toxicosis due to excess TSH. Even perineoplastic syndromes, where tumors elsewhere in the body, like lung cancers, ectopically secrete hormones, can cause pituitary hypersecretion by releasing CRH or GHRH, effectively mimicking a hypothalamic signal from a distant source.
Let's delve a bit deeper into precocious puberty and the specific role of these hypothalamic hamartomas.
Right. While true familial idiopathic sexual precocity, early puberty with no identifiable cause running in families, is rare, many cases previously labeled as idiopathic are now understood to be linked to small hypothalamic hamartomas, detectable with modern MRI imaging. As mentioned, these are tumor-like collections of essentially normal nerve tissue, but located abnormally, often attached to or near the floor of the third ventricle or the tuber cinereum. They can cause precocious puberty. There are generally two types: parahypothalamic, which are pedunculated or stalked, and intrahypothalamic, which are sessile or directly attached to the hypothalamus. The latter type tends to be larger and can distort the third ventricle, often being accompanied by neurological symptoms like seizures, especially gelastic or laughing seizures, cognitive decline, and developmental delays. Precocious puberty occurs when the hamartoma, which contains functional GnRH neurons, makes connections with the median eminence portal system, allowing it to act as an accessory hypothalamus with its own intrinsic, unregulated GnRH pulse generator, starting puberty too early. Early signs can include those characteristic gelastic seizures, brief, repetitive, stereotyped attacks of laughter without mirth. Management often involves using long-acting GnRH agonists to suppress the pituitary's response to the hamartoma's GnRH, effectively pausing puberty. Surgical approaches might be considered for sessile hamartomas associated with intractable seizures, but surgery in this delicate area carries significant risks, including potential hypothalamic obesity, diabetes insipidus, and other pituitary hormone deficiencies.
What about tumors originating in the pineal gland itself, that once mysterious seat of the soul? Are they common?
Pineal gland tumors are actually quite rare, accounting for only about 1% of all intracranial neoplasms, or brain tumors, in adults, though slightly more common in children. They typically appear as central midline masses on MRI, often compressing the aqueduct of Sylvius and causing hydrocephalus, or fluid buildup in the brain. They can lead to a variety of neurological abnormalities, most classically paranovus syndrome, a paralysis of upward gaze due to pressure on the midbrain tectum. While pinealocytes, the main cells of the pineal gland, can give rise to tumors, pineocytomas, pineoblastomas, the most common tumors found in the pineal region are actually germinomas. These originate from misplaced embryonic germ cells and can also appear primarily in the anterior hypothalamus or the floor of the third ventricle. Supracellular germinomas are often associated with a triad of diabetes insipidus, pituitary insufficiency, and visual abnormalities due to their location. These tumors can spread locally and sometimes metastasize via the cerebrospinal fluid. Precocious puberty is an unusual manifestation of pineal gland disease itself, typically occurring only if a tumor puts pressure on the nearby hypothalamus, or, very rarely, if a specific type of tumor, like a choriocarcinoma, secretes human chorionic gonadotropin, HCG, which can weakly mimic LH action. Management is complex and depends heavily on the tumor type, involving surgery, radiation, or chemotherapy.
Can the hypothalamus also be the source of too much growth hormone, leading to conditions like gigantism or acromegaly?
Yes, hypothalamic causes of GH excess exist, though they are rarer than pituitary adenomas secreting GH. The dancephalic syndrome, typically seen in infants and young children with tumors located near the floor of the third ventricle, often low-grade gliomas or astrocytomas, can cause profound cachexia, severe wasting, and malnutrition, often paradoxically accompanied by elevated GH levels and abnormal GH responses to stimuli. The GH hypersecretion might result from direct hypothalamic abnormality caused by the tumor, or possibly secondary to the severe malnutrition itself. These children often present with a strikingly alert, almost euphoric appearance despite their severe emaciation. Most cases are due to chiasmatic hypothalamic gliomas. More recently, a specific genetic condition called X-linked acrogenism, XLAG, has been identified as a cause of very early onset GH excess, often starting in infancy. This is due to microduplications on the X chromosome involving the GPR101 gene, which encodes an orphan G protein-coupled receptor. Affected individuals often have pituitary adenomas or hyperplasia, overgrowth, of GH-producing cells, frequently accompanied by elevated GHRH levels, suggesting the primary defect might involve GHRH regulation or signaling, leading to the excessive growth.
What are some of the broader, more general consequences of significant hypothalamic damage beyond specific hormone issues?
Patients with significant hypothalamic damage can experience a disturbing range of severe thermoregulatory problems. This can include persistent hyperthermia, high temperature, hypothermia low temperature, unexplained and erratic temperature fluctuations, or even poikilothermy, a profound inability to regulate body temperature at all, causing their temperature to passively follow the environment. Disturbances of sweating, loss of bladder or bowel sphincter control, and a specific type of seizure originating from this area called diencephalic epilepsy can also occur. Crucially, hypothalamic damage, particularly involving the mammillary bodies or their connections, often causes significant impairment of recent memory consolidation, making it hard to form new memories. This is believed to be due to damage to the vital mammillothalamic pathways. Severe memory loss, often coupled with morbid obesity and profound personality changes, ranging from apathy and loss of concentration to aggressive antisocial behavior and severe uncontrollable food craving, can result from supracellular extension of pituitary tumors, compressing the hypothalamus, radiation therapy directed at this area, or surgical damage during attempts to remove nearby lesions. This really highlights the incredibly delicate balance involved in treating conditions in this region. Slowly growing lesions might produce minimal disturbance initially as the brain compensates, but even seemingly limited surgery or intervention can cause striking and often irreversible functional abnormalities due to the density of critical functions packed into this small area.
Given how intimately the hypothalamus is tied to our circadian rhythms and sleep regulation, sleep disorders must be a major manifestation when it's dysfunctional.
Indeed they are. Narcolepsy, particularly narcolepsy with cataplexy, that sudden loss of muscle tone triggered by strong emotions, has been profoundly linked to hypothalamic dysfunction. Major breakthroughs came from studies in narcoleptic dogs and later mice, which identified mutations in the hypocretin orexin receptor 2 gene and the orexin hypocretin peptide precursor gene itself as the direct causes of narcolepsy in these animals. This groundbreaking work definitively established the orexin system, originating exclusively in the lateral hypothalamus, as a major indispensable component of sleep-wake modulating neural circuits. In humans with typical narcolepsy, type 1 with cataplexy, there's a profound and selective loss of these orexin hypocretin neurons in the lateral hypothalamus. Most spontaneous narcolepsy with cataplexy is now thought to result from an autoimmune process that specifically targets and destroys these neurons, likely triggered by an infection in genetically susceptible individuals. Consequently, the absence or extremely low level of immunoreactive orexin hypocretin in the cerebrospinal fluid, CSF, has become a highly sensitive and specific diagnostic test for narcolepsy type 1.
What about other, perhaps less common, but still significant sleep disorders that might be linked to hypothalamic dysfunction?
Klein-Levin syndrome is a fascinating, rare periodic sleep disorder. It often affects adolescent males and is characterized by recurring episodes of severe hypersomnia sleeping for most of the day, accompanied by cognitive disturbances, hyperphagia, compulsive overeating, and sometimes hypersexuality or other behavioral changes. Between episodes, individuals typically return to completely normal behavior. Unlike narcolepsy, CSF orexin levels are usually normal in Klein-Levin syndrome. Its exact etiology remains unknown, though autoimmune or inflammatory theories, potentially triggered by infections, and some genetic links, like variants in the LMOD3 gene, are being actively explored. Prader-Willi syndrome, a genetic disorder primarily known for neonatal hypotonia, hypogonadism, hyperphagia leading to obesity and cognitive impairment, is also frequently associated with significant hypersomnia and disrupted sleep architecture. Additionally, very rare conditions like rapid onset obesity with hypothalamic dysfunction, hypoventilation, and autonomic dysregulation, or AHEAD, can occur in children. This devastating condition involves abrupt onset of obesity, impaired breathing control, hypoventilation, autonomic nervous system instability leading to problems with heart rate, blood pressure, and body temperature regulation, often accompanied by endocrine abnormalities like GH deficiency and central hypothyroidism, clearly pointing to widespread hypothalamic failure. In the elderly, a proposed condition termed Serklon's syndrome, circadian clock neuronal senile atrophy, potentially involving age-related degeneration or senescence of the SCN, might contribute to the severely fragmented sleep patterns often seen in advanced age.
Finally, let's try to connect the hypothalamus to broader neuropsychiatric disorders, going beyond the purely endocrine or primary sleep disorders.
Right. The hypothalamus's well-established role in fundamental drives like eating, and thus its involvement in eating disorders like anorexia nervosa, is certainly widely recognized. But there's growing evidence implicating hypothalamic dysfunction or dysregulation in complex neuropsychiatric conditions like schizophrenia and major depressive illness as well. For example, genome-wide association studies, GWAS, looking for susceptibility genes for schizophrenia, have identified the hypothalamus as one of the major brain regions where these risk genes are predominantly expressed. These genes often affect neurotransmitter receptors, ion channels, and neurodevelopmental processes, suggesting hypothalamic circuits might be particularly vulnerable. Enlargement of the third ventricle, which sits within the hypothalamus, is a finding sometimes observed in imaging studies of both schizophrenia and major depressive illness, potentially reflecting a loss of surrounding periventricular gray matter. This naturally leads us to ask, What about specific peptide systems known to be regulated by the hypothalamus? Dysregulation of the oxytocin and orexin systems has been implicated in mood and psychotic disorders. Polymorphisms in the oxytocin receptor gene have been identified as potential risk factors in schizophrenia and major depressive disorder. And some preliminary studies have shown potential therapeutic benefits from intranasal oxytocin administration in these conditions, although results are mixed. Similarly, oxytocin has been investigated in autism spectrum disorder, with some reports of reduced plasma levels and potential improvements in social interaction measures after intranasal administration, though again, more research is needed. Orexin deficiency or receptor downregulation has been observed in animal models of depression and might contribute to the disrupted sleep patterns common in human depression. Orexin administration has shown antidepressant-like effects in some preclinical studies. The potential antidepressant actions of kisspeptin and emerging evidence linking peptides like somatostatin and nesfatin-1 to major depressive disorder further indicate that there is likely much more to be learned about the hypothalamus's profound and far-reaching role in the pathophysiology of neuropsychiatric disease. #outro.
Wow. That was an absolutely incredible deep dive into the hypothalamus. I mean, from its ancient recognition way back with Galen to the cutting-edge genetic studies and therapeutic applications we're seeing today, we've really seen how this small but truly mighty region acts as the conductor of your body's most fundamental processes. It's just astonishing how much it orchestrates.
It really has been quite a journey. We've navigated its historical understanding, dissected its precise neural control over hormone secretion, explored the incredibly intricate hypothalamic pituitary unit, and shed light on crucial homeostatic functions like thermoregulation and our sleep-wake cycles. And importantly, we've also seen the profound and sometimes devastating impact when this delicate balance is disrupted, leading to that wide range of neuroendocrine diseases from growth disorders and reproductive issues to sleep disturbances and even links to complex neuropsychiatric conditions.
Yeah, this deep dive has truly shown us that mastering this information isn't just about understanding complex biology, although that's part of it. It's really about appreciating the incredible, intricate interconnectedness that underpins your health and your well-being every single moment of every single day. The level of detail required to truly master this subject, like the elite professionals strive to, just reveals how much precision is constantly at play within us.
So what does all this complex detail mean for you, the listener, in practical terms? Well, perhaps next time you feel that subtle shift in your energy level or the natural pull towards sleep, or even just the simple pang of hunger, maybe take a moment to consider the incredible unseen symphony being orchestrated by your hypothalamus. It really raises an important forward-looking question. How might a deeper, more refined understanding of these precise neural circuits and hormonal controls, getting down to the level we've discussed today, unlock even more personalized, perhaps even groundbreaking, approaches to health, well-being, and treating disease in the future? The possibilities, I think, are truly immense.
Thank you so much for joining us on this deep dive into the hypothalamus. Until next time, stay curious and keep exploring the amazing world within you.